You know, when I think about doing honors thesis research, there's no road when you start honors thesis research. It's really the end of the road, and there's this big yellow sign. It says wilderness ahead. Proceed at your own peril. Proceed with caution. And if you're lucky, be prepared to make some exciting discoveries. So it's a really daunting challenge, but it's really one that's at the heart of what we're trying to do at Stanford in terms of transforming you from a student to a scholar and from a student to a teacher. And I think about today as a kind of a rite of passage. Today's a rite of passage in which you become that teacher. But fortunately, you're not doing that alone. You have very sage and wise mentors, graduate students, postdocs, faculty, staff who are advising along the way. You may have called your parents or brothers or sisters a time or two and complained or even cried about how difficult this endeavor is. And you certainly have reached out to friends. So we're doing this in good company. And when I look at the topics that are being discussed today, we have everything from water resources to various ecological impacts, or I should say climate change impacts on ecology. There's really a broad diversity that reflects the research interests of the school, but also the research interests that you've owned and that you've taken a special part of and delved into them. So they're really an opportunity for you to express your desire for your own sense of inquiry and to own these projects. So when I think about what we're doing today, I just want to celebrate what it is that you're doing, this transformation from scholar, or from student to scholar, from scholar to teacher, and welcome you all. I'm really looking forward with great anticipation and excitement from what we all have to learn today. So with those words, I'll welcome our first speaker, Radis, who's done some really interesting work up in southeastern Alaska. The title of her talk is Understory Plant Response to Yellow-Cedar Decline, an Exploration of Nutrient Uptake and Availability.
Thank you, Richard. So for those of you guys who don't know, my name is Corey, and I did my Earth Systems thesis project on yellow-cedar decline in southeast Alaska. So before I begin, has anybody here heard of yellow-cedar decline before? And has anybody heard of the bark beetle phenomenon? So these two forest mortality phenomena are similar, and then they show how a small climatic shift can have major impacts on forest ecosystems, but different phenomena. I'll give you guys an introduction to my project, talk a little bit about what yellow-cedar decline is, my mentors, and my methods, what I did, field work, lab work, and finally what we found, results and implications. The yellow-cedar trees are a common coniferous tree that grow throughout the Pacific Northwest from California up to British Columbia and Alaska. They have economic, ecological, and cultural value. They're a prized timber product. Indigenous people have used yellow-cedar trees for years to build canoes, totem poles like this one. They even eat some parts of the bark and the leaves for medicinal purposes. And of course, yellow-cedar trees host great habitat for a number of species and host diverse forest ecosystems. Unfortunately, over the last century, yellow-cedar trees have been experiencing decline, basically since the onset of the Industrial Revolution, and it's thought to have been caused by climate change. I'm not going to get too into the causes of yellow-cedar decline, because that's not what I focused on for my research, but just a basic overview. What the leading hypothesis of what's happening is that yellow-cedar trees have these fine roots that they send up to the surface in the springtime to get nutrients from spring runoff. And historically, when this happened, the roots would be covered by a layer of snow that would insulate them. However, in recent years, snow has been melting earlier in the spring, and so these fine roots are exposed to freezing temperatures, undergo a process called sudden frost death, and die. So the map on the left shows the range of yellow-cedar trees, with the red parts zoomed in up top, showing where yellow-cedar decline is happening. And the picture on the right is a picture of yellow-cedar decline. And it's funny because it's interesting to learn about this, but when you actually get up there, I remember my first ferry ride up through southeast Alaska, looking up at the hills, and the forest is just brown. It's pretty hot. So I worked with Lauren Oakes. Lauren is going into her 5th year in the E-HYPER program here, and the interdisciplinary program in the environment and resources. And Lauren is studying what happens next. What's going to replace these dying forests? She's looking at both the social and the ecological implications of yellow-cedar decline. So for my project, well, to learn that, I designed a project that would ultimately supplement her dissertation.
Other people that I've been working with are Jennifer Johnson, who is was a PhD student over at Carnegie. She since graduated and now works in Tucson, but she guided me through the lab work. Joe Berry and Chris Field are two other researchers who many of you guys are probably familiar with, researchers at Carnegie who helped me out with the plant biogeochemistry part of the project. And then Paul Hennon and Dave Diamore are researchers in Southeast Alaska. They work at the Pacific Northwest Research Station. And they are kind of the yellow-cedar guys. They're the ones who came up with the whole hypothesis of how the trees are dying. So for my project, I looked at the understory response to yellow-cedar decline. So as yellow cedar dies, what's happening to these understory plants? In particular, we asked the question, does the nutrient flow associated with yellow cedar decline have an impact on understory shrubs and forbs? We hypothesized that as yellow cedars die, they stop taking up nutrients and increase the pool of nutrients in soils of dead yellow cedar forests, making more nutrients available for understory plants. To do this, we chose 4 understory species: Alaskan blueberry, bunchberry, western hemlock saplings, and deer fern. We chose these 4 plants because they represent a range of plant taxa, and they're also key forage for wildlife in the area, in particular Sitka, black-tailed deer, and bird species. So the game plan that we decided on, this was my sophomore spring, was to go up to Southeast Alaska in the summer, collect leaf samples from these plants from both healthy and dead yellow cedar forests, dry the leaf samples, bring them back to Stanford campus, analyze them for their nutrient concentrations, and then using Lauren's biomass estimates, convert these nutrient concentrations into nutrient pools. So when I say nutrient pools, I mean, for example, milligrams of nitrogen per meter square of ground. And by comparing nutrient pools in healthy forests and dead forests, we can look at how the nutrient availability for wildlife changes as yellow cedar trees die. So our study area was in West Chichagof-Yakobi Wilderness. It's about a 2-hour boat ride north of Sitka in Southeast Alaska. It's very, very remote, and Lauren had been doing fieldwork there for a few years. Being that it's very remote, and that's a good thing for fieldwork because it is basically untouched by humans. Lauren had established 4, we called forest types. They're basically levels of yellow cedar mortality throughout our study area. These included healthy forests that had not been affected by yellow cedar decline at all, as sort of the control, and then 3 levels of mortality, with the oldest level being trees that have died up to 100 years. So for example, this picture in the back is probably a mid-mortality forest. So the summer of 2012, that was the summer after my sophomore year, we went up to Southeast Alaska for fieldwork. And these are just a few pictures from fieldwork. Fieldwork was challenging for a lot of reasons. The West Chichagof-Yakobi Wilderness is actually one of the places with the highest concentration of grizzly bears anywhere in the world. So this was not taken lightly. We were constantly alert. Up in the left, that's grizzly bear training that we went through in Sitka before going. And we were constantly prepared, didn't run into any problems. The other challenge is that Southeast Alaska is a rainforest, so we would go days without seeing the sunlight. And for field research, this makes it especially difficult. We had to, be very prepared, have all our data sheets. We learned that, write in the rain paper isn't actually waterproof. We used scuba diving paper, and after a day, that, would start to disintegrate. But we spent weeks getting things together, all our stuff, and it was a success. It worked out. A few more pictures from fieldwork, collected samples, and after about 20 days in the field, returned to Juneau to dry them at the Forestry Sciences Lab, where Paul and Dave work, and then shipped those samples back to Carnegie for analysis. So then the winter and spring of my junior year, I worked on analyzing these understory leaves. We first ground the leaves up into a fine powder using this machine on the left, which is called a ball mill. It's a pretty incredible machine that someone out at Carnegie actually made themselves. And you basically load the samples up, and then it shakes really hard until the leaves are a fine powder. It's awesome. And then use 2 lab processes to get the nutrient concentrations. The first one was the ICP, oh yes, actually right here in EM1. So I digested the dry leaf material with nitric and hydrochloric acid. And then with the ICP, found nutrient concentrations. We were in particular interested in calcium and phosphorus, but got a whole spreadsheet of numbers to play with. The second process is this Carlo-Erba isotope ratio mass spectrometer machine, which is over in Carnegie. And to prepare samples for this, you foil ball them. Another process I'd never done before, but learned a ton. And this was for carbon, nitrogen, and the stable isotopes. So what did we find? We found actually pretty close to what we expected, which was really, really exciting. This figure on the left here shows nutrient pools, carbon, phosphorus, and nitrogen nutrient pools from healthy forests to old mortality forests. And there's a clear increase in nutrient availability in the forests that have experienced yellow-cedar decline. This is largely driven by a change in biomass, so the fact that there's actually more understory plants in old mortality forests. However, we found that increasing nutrient concentrations also contributed to this figure. So plants are both more abundant and more nutritious in forests affected by yellow-cedar decline, for the most part. So here's another example of the nutrient concentrations. This is just nitrogen in blueberry plants. So you can see on the left here, that's the nitrogen concentration in a healthy forest, and the other 3 are nitrogen concentrations in forests affected by yellow-cedar decline. So it's really interesting that as these yellow-cedar trees die, the nitrogen dynamic in the forest is totally changed. So what does this mean for wildlife? What we're suggesting is that in forests affected by decline, the forage will be both more, as I said, abundant and nutritious for wildlife, in particular for the Sitka black-tailed deer and bird species. And forest mortality is not a phenomenon that is unique to Southeast Alaska. Forest mortality phenomena are actually occurring on all 6 plant-covered continents right now. And so understanding how these forests evolve and how we can conserve or manage them in the face of climate change is really interesting. One idea that I discussed in my paper is if the carrying capacity of deer, for example, is really increasing in dead forests, maybe we should shift deer hunting to dead yellow-cedar forests and preserve live yellow-cedar forests. Or there's a process called salvage harvesting, where you can actually harvest these dead yellow-cedar trees. They preserve their timber qualities for almost 80 years after they've actually died. So harvesting dead forests instead of harvesting live forests. So overall, I learned a ton from this project, had an amazing time. Looking back, I definitely think of it as one of the most valuable components of my undergraduate, my understory education, my undergraduate education. And I'm grateful to my mentors, to Richard for helping me through the assessor process, and the Earth Systems faculty, and my friends. I hope to continue this project through the summer with Lauren a little bit, just working to fine-tune our results and hopefully make it back to Southeast Alaska someday too. Thank you. Love to take any questions if we have time.
Yes, I'm just going to repeat the questions, so go ahead.
I'm wondering if there's any work with invasive species, if the increased nutrients has caused any issues with invasion of shrubs maybe that you didn't look at, but that are taking over where the cedar is dying.
So the question is, will there be an impact on invasive species?
That's a great question. As far as I know, there has not been research looking into invasives in this area. I would guess that one reason is because the West Chichagof where we're studying is so remote that there aren't too many invasives there. However, I would imagine that yellow cedar forests closer to civilization where invasives are more of a problem, it'd be really interesting if there's more nutrients that invasives could take over. So hasn't been any research, but it would be really interesting to look at. Yes, Jesse.
So you're saying that the plants have more nutrients in the understory after die-off. Do you willing to speculate whether this is because the canopy is opening up and there's more light so they can more actively get the nutrients, or that the depth of the canopy is actually providing them to cool? Do you have like soil samples that could say like there's a change in the soil pools perhaps?
So the question relates to the cause and effect relationships between increased nutrient content in the leaves, and whether that's driven by more sunlight or by more direct transfer of nutrients from the dying yellow cedar to the understory.
So this is a big question, and that's definitely, I don't want to say a hole in the study, but how can we tell if it's caused from light or caused from soil? One piece of evidence that we found that shows that plants are actually taken up more not because of light is that the N15 values change, showing that the whole nitrogen dynamic is changing. So it's not just from light, it's like they're taking up this different nitrogen source. So that's our key tip that there is something going on beyond light. I do think that light contributes in a big way though.
Norm?
I noticed that some of the trees were still alive even though you had lots of dead ones. And are those a different species, or are those just, because it seems if some of the cedars survive with natural selection, once their seeds got spread around, that the forest could partly recover from this.
So the question relates to what's the cause of only some trees dying in these areas that have experienced a lot of mortality.
Yeah, that's another great question. I don't know for sure. I think these forests are mixed forests, so they're yellow cedar trees, but also western hemlock and Sitka spruce. So the live trees, especially in those pictures, might have just been other species that aren't affected by decline. One thing I didn't mention is that a reason that just yellow cedar trees are affected by decline is because of their fine roots. Western hemlocks and other conifer species in the area don't have this adaptation, and so they're not experiencing decline. So I would guess that all yellow cedar trees in an area would die, while other ones would maybe take over. But yes.
Any more questions for Corey? Corey, I have a couple of questions.
I'm ready.
Really great presentation.
Thank you.
Really interesting. So I noticed that in your results you were looking at western hemlock, but we didn't see any data from that. Is there a reason for that? Or maybe I missed that.
Let's see. This one or the one before?
I think it was that one. Is a western hemlock included in that?
This one I didn't, that's a great point, is I did not include western hemlock in this figure. This is the cumulative nutrient pools of the other three species. So blueberry, bunchberry, and deer fern. I didn't include western hemlock because it's not relevant to bird species. And in my thesis, I wanted to have one figure that could speak to both bird and deer. So that's the only reason. Western hemlock did have a similar trend, but yeah.
Why didn't you do Devil's Club?
Devil's Club, because it's so difficult to sample.
Fair enough.
Good answer. I have several questions, so we have time. So
Yes.
One more question. So what do you speculate will be the long-term impact of this yellow cedar decline? Are you going to see more trees dying? What are going to be the successional species? What will replace the role of the cedar? Will it be the western hemlock in these forests?
That's the big question that Lauren's probing at right now. So what is going to replace yellow cedar trees? It's really interesting, and this is another thing I talked about in my discussion is seeing the shift in resources, from yellow cedar trees, which are valuable in so many ways, as I explained, to understory species and other types of conifers, which in and of themselves have value. So I think that, my personal speculation is that maybe right after yellow cedar trees die, there's this understory boom, and eventually another conifer species will take over. Western hemlock seems like the big one right now. But there's sort of a forest migration phenomenon where maybe yellow cedar are moving north to colder areas, and species that can tolerate those warmer temperatures move up to take their place.
All right, thank you for a great presentation.
Thank you.
Our next speaker is Caleb Kruse, and a lot of you know Caleb really well, but some of you may not know that this project was really born out of some interesting work he did with a blender when he was still in high school. So you might want to ask Caleb about his early work and how that's connected at the end of the talk. The title of Caleb's talk is Symbiont Production of Superoxide in Thermal Coral Bleaching Independent of Photosynthetic Activity.
All right, thanks, Richard. So today I'm going to be focusing on coral bleaching and kind of some of the mechanisms of how it works and a possible new mechanism for the bleaching process. I think all of you are pretty familiar with bleaching in general, but just to make sure that everyone's on the same page, just go through it real quickly. So you start off with a really vibrant, healthy reef like this. And reefs like this are just huge biodiversity hotspots. They host anywhere from 500,000 to 10 million different species, a wide range. But you know, a lot of different species, 32 of the 34 animal phyla, real just valuable spots in the world. But you get any sort of perturbation of the environmental conditions, a change in temperature most often, change in light intensity, change in chemical balance, or even bacterial or viral disease, and a healthy coral reef that looks like this quickly declines into a dead, bleached coral reef. So coral bleaching is really a prominent disease or phenomenon that can wipe out entire reefs in the matter of just a couple weeks. Once a coral has bleached after a severe bleaching event, there's less than 10% chance of survival. So we've seen real large-scale bleaching events be a big problem, most notably the 1998 bleaching event that devastated a lot of the reefs in Australia. But it's increasing in severity and frequency as the water temperatures continue to warm. So a little bit more about bleaching. What's actually going on here? We saw that the coral died and became white, but what's actually happening? Well, it's all based on the fact that corals and anemones are symbiotic most often. And they're symbiotic with a small dinoflagellate or algae called Symbiodinium. And this algae lives inside the tissues and provides necessary nutrients through the byproducts of photosynthesis. So in this image, you see an anemone here, and when you look at it with fluorescence microscopy, it's a little hard to see here, but all the red in this image is due to fluorescence of chlorophyll. And so you see that the algae is living throughout the host and is really a crucial part of that system. But then when you get a stress, the host, the symbiont is lost. And this results in death of the coral oftentimes or anemone because the symbiont produces or provides up to 90% of the host nutrient source. And so here once again in the fluorescence microscopy, you can see just when the symbiont is lost during the bleaching process. So what would cause this? Because bleaching is a very counterintuitive response. It seems like there's a stress and the last thing that a coral would want to do is kick out its prime nutrient source. It just doesn't make kind of intuitive sense. So there has to be something deeper going on. And so in the mid-90s, we came up with this idea that was based around a group of compounds called reactive oxygen species. And reactive oxygen species take the form of singlet oxygen, the superoxide ion, hydroxyl ion, and hydrogen peroxide. And these compounds are incredibly damaging within the cell. They can oxidize lipids, tissues, and even DNA. And so they're just really, if they can't be contained with antioxidants or defense mechanisms, they can really wreak havoc. And so the idea was that we saw that reactive oxygen species concentrations were higher in corals that had bleached. So the thought was, well, maybe the symbiont is producing reactive oxygen species, and so the coral either suffers with oxidative stress, or it kicks out its symbiont and reduces that oxidative stress, but then starves. So how does this actually happen? Well, this is the model of bleaching that we've been operating under for almost 20 years now. It was developed by Michael Lesser, and it's the idea of photoinhibition. So here, it places the demise of the symbiosis on the symbiont's photosynthetic machinery. So this is healthy photosynthesis right here, and you see that you've got a nice transfer of electrons, ultimately producing ATP. But the problem is when you have a phenomenon called photoinhibition. When light levels increase past a certain level, the D1 protein of photosystem 2 is degraded, and it's degraded at a faster rate than it can be regenerated. And what this means is the reaction center is still functioning perfectly. It's absorbing light and converting this to high energy electrons. But since it can't pass these high energy electrons down the electron transport chain, these electrons are released into the thylakoid membrane or cytosol and end up oxidizing oxygen, producing those harmful reactive oxygen species. So Lesser's idea was you have this UV light stress, and the symbiont produces reactive oxygen species, so that's why the coral is kicking out its symbiont. But there's a couple problems with this theory. The first is the symbiont is still photosynthetically capable after being expelled from the host. In measurements of FV over FM, which characterizes photosynthetic capacity, it appears that they still have the ability to photosynthesize, and that would not hold with this idea of photoinhibition. Second, it doesn't take into account the role of temperature. This is entirely light dependent and explains nothing about how coral bleaching would be instigated by an increase in temperature, which is really important because that's mainly what we see in the natural environment. So this led to an experiment last year on dark bleaching, and it really changed the way that we think about coral bleaching. Two students here at Stanford, Dimitri Tollider and Francois Seneca, did an experiment where they said, hey, we're interested in the role of temperature. And so they did an experiment where they had corals and anemones that were up at 34 degrees, a high temperature in both the light and the dark. And they compared the rates at which these two groups bleached. And if what they saw is that the rates of bleaching in both the light and the dark were equivalent. And what this indicates is if photosynthetic degradation was the culprit for this breakdown in symbiosis, then the corals in the dark with just a high temperature probably shouldn't have bleached. So this is a very interesting finding. It really changes how we think about bleaching, but it left us without a mechanism. So there was no mechanism, and we didn't know whether reactive oxygen species are still playing a role in coral bleaching even in the dark. So that's where my research comes in. It's where we see that maybe the model that we've been using for bleaching for a while isn't quite correct, but don't really know where to move forward from here. So my experiment had two purposes. First was to determine whether reactive oxygen species are produced during the dark bleaching process. Perhaps ROS are only produced in the light, and there's an entirely different bleaching process in the dark. My second purpose was to find the source of reactive oxygen species production in the process of thermal bleaching. So independent of light, I want to see where ROS are being produced if they are. So how did I do this? Well, I had a really nifty tool, and that's the Aptasia anemone model system. And at first glance, you might be a little bit taken aback, like, wait a minute, we're talking about coral bleaching here. What are we doing with anemones? Well, anemones are just a really nice tool that were developed in the Pringle lab where I had conducted this research. And these anemones are really beneficial because they're very hardy. They're symbiotic with the same algae that's symbiotic with coral. They reproduce asexually, so we can maintain a clonal strain, and just really easy to work with. So that's a picture of one of the Aptasia anemones under fluorescence, and this is what they look like more in natural color. They're about 1 to 2 centimeters across and very hardy. And so the main differences between one of these anemones and a coral is they don't produce a calcium carbonate skeleton, and they're a single polyp rather than a polyp in a larger community structure. But really a wonderful model system for us to get at more of the molecular side of coral bleaching. One other prominent feature of this system is they can be maintained aposymbiotically. So a coral, if it had none of its symbiont, would die almost immediately. But in the Aptasia model system, if we feed the anemones that are don't have their symbiont, they can stay alive, and that allows us to do some really interesting comparisons. Next, a technique I used, I'm very interested in reactive oxygen species production. And specifically, I looked at the superoxide production. Reactive oxygen species are very difficult to measure due to their reactive nature. And there's very few molecular probes that can measure them very accurately. But I used this probe, nitroblue tetrazolium, which it was commonly used for looking at the ability for white blood cells in humans to produce the necessary ROS to kill an invading cell. I applied the technique to be used on Aiptasia anemones, and the way it works is you start out with a soluble brownish, light brown yellow solution, and then as soon as it accepts 2 electrons from superoxide, then it turns into a precipitate, and it's very dark purple, and I could then quantify this. So the experiment had 2 experimental groups. There was the symbiotic and aposymbiotic anemones, and within these 2 groups there were 4 treatments. There was the negative control, where the anemones were kept at a non-stressful temperature and not supplemented with MBT. I just wanted an untouched sample set to compare against. There was the positive control, maintained at a non-stressful temperature, but included NBT, so I could monitor a basal level production of superoxide. And then 2 experimental groups of known bleaching temperature, 34 degrees, with NBT added in both the light and in the dark. And this would allow me to do some pretty interesting comparisons. So to get a sense of what I was actually kind of looking at here, here's a picture of an anemone in the negative control. As you see, it's kind of its typical brownish, translucent color. But then once you add NBT to the solution, this anemone was kept at 34 degrees with NBT added. You see that it's got a very dark purple color. And so the way that I was characterizing superoxide production was to use this dark purple color as a proxy for that production. And then I could quantify that. So it took a little tricks in isolating it, but I ended up getting a method that seemed like it was working. Just a sanity check to make sure that this NBT compound was actually working. I wanted to compare samples treated with NBT with no superoxide present. And so the way that I did this is I added superoxide dismutase, an enzyme that neutralizes superoxide, to a sample and treated it with NBT. That essentially brings the superoxide concentration down to 0. And you see that the absorbance there was very low. In comparison, when no superoxide dismutase was added, there's much higher concentration. So that indicates that the assay was working and could characterize at least relative concentrations of superoxide production. So now to the results, the most exciting part of this and what it means. So in this graph, we see on the y-axis there's absorbance per milligram of protein. What this is really looking at is the relative concentration of superoxide production in the different groups. The black bars here represent the symbiotic anemones, gray is aposymbiotic, and then the treatments are listed. And what we see is there's only one dominant trend. It's the fact that the anemones, the symbiotic anemones, at 34 degrees in both the light and the dark, produced a high level of superoxide. In all other samples, the production of superoxide was indistinguishable. So the only place where we see this really increased superoxide production is in symbiotic anemones at high temperatures. And this is really interesting. The way that the experiment was set up was so that we could kind of tease apart various hypotheses of where the superoxide was being produced. So suppose we go with Lesser's idea of photoinhibition. That would show that what we would expect to see there is only a spike at 34 degrees and in the light, because that's where photoinhibition would take place. If reactive oxygen species were alternatively, if they were produced by the host rather than the symbiont, we wouldn't see this dependent on symbiotic or aposymbiotic. We would see just elevated reactive oxygen species production in both 34-degree groups. What we do see, this trend where only symbiotic anemones at high temperatures producing high levels of superoxide, indicates that the superoxide production is coming from the symbiont, but it's independent of photosynthetic processes. We don't see a significant difference between the 2 groups in the light and the dark, showing that it's probably coming from somewhere else. So more research is needed on this idea, but an obvious culprit would be the mitochondria. The mitochondria in almost all organisms is basically reactive oxygen species are produced either by UV or the mitochondria. UV radiation or mitochondria. So mitochondria in the symbiont is a natural place to look. When you increase temperature by 7 degrees, you're getting an approximate 65% increase in algal metabolic rates. And so it would just make sense that the algal mitochondria now is ramping up its metabolic rate and producing more of these reactive oxygen species. So to conclude, we see that ROS are produced by the symbiont, but independent of photosynthetic activity. This is kind of a shift on our old model of bleaching, but could really provide some insights. And why this is so important is it resolves a lot of the problems in the field. So first of all, it provides an explanation for the expulsion paradox, that idea of why is the coral expelling its symbiont, which is necessary to keep it alive? Well, the symbiont is producing something that's toxic to the coral still. It also explains the temperature dependence of bleaching, because the mitochondria metabolic rates would increase with temperature, and that would lead to the increase in production of ROS. And finally, it provides an explanation for the dark bleaching phenomenon, how corals are bleaching in the dark. So it really could be beneficial. In future work, I want to look more into the temperature dependence of algal metabolic rates, and also target more, find a way to actually specifically show that the reactive oxygen species are being produced in the mitochondria. So finally, I'd like to thank the Pringle Lab. They really were awesome, as Richard alluded to. I've been thinking about coral bleaching for a long time, and it's something that I like to study. And they really took me in my junior year and just did everything possible to make this research take place. Especially Corey was an ideal mentor of not meddling too much in my experiments, but anytime I had a question, being just ready and able to answer it. So thanks.
All right, questions for Caleb?
Yeah.
So I have a question about the symbionts. So
Yeah.
the coral or the anemones, they eject these symbionts. They're in this different state of, emitting all of this ROS. What happens to the symbionts? Do they die? Are they able to find another host? What's happening in the ecosystem?
So there's,
So let me just restate the question. I'm only doing this so we can pick it up on the mic. So the question is, when these coral reject their symbionts, what happens to them?
yeah, it's a good question. So I think in an ecological sense, we don't really know. There have been some free-living Symbiodinium strains, but we don't know if they're the same strains that are present in the corals. But there's also the idea of repopulation. So corals can survive after a bleaching event. Depends on the severity. And we don't really know whether corals and anemones have the ability to take in Symbiodinium from their external environment and then it repopulates, or whether a small amount of that Symbiodinium remains inside the host and then it can just regrow. But generally, there is Symbiodinium in the water because the offspring of corals and anemones have to populate somehow so they can be recolonized.
Nora?
We have peroxidase in our body to get rid of these oxidizing compounds, and we can also take 37 degrees C and a bit hotter if we get the flu. It just always amazed me in the geological record that reef-forming organisms seem to just be so singularly unfit. And then, is there kind of a larger reason for that?
Yeah. Do you want to repeat it?
Sure. Why is it that when you look throughout the geological record, it appears that coral-forming or reef-forming organisms seem to be so unfit? How come this hasn't been winnowed out at this point?
I don't know. It's a good question. I think it has to do with, this is speculation here, but the fact that symbiosis is a really tentative phenomenon. So both partners have to be working perfectly in tandem. So when you have two organisms, one organism living inside of the other to make the system function, any small perturbation can result in, a collapse. But throughout the geological record, I don't know.
Things like the rudous clams that dominated the Cretaceous and actually went extinct before the main extinction.
Yeah, I'm not really sure. But they're doing all right. They're still around today. I hope they can pull through now, but I'm not sure.
We have time for maybe one more question for Caleb. Well, Caleb, I have a question for you. So I'm really intrigued by what you learned here. And it seems like it's going to be pushing the research to just looking at not the host, but what is being hosted independently. Do you have an idea of a model organism that you might want? Is that correct?
Sort of. I think it probably would be more using the Aptasia model system, continuing to work with it in order to pinpoint this at longer time points in the bleaching process, and then also localizing that ROS production to the mitochondria within the algal cells. But I think with cultured strains of algae and then also the Aptasium model system, it's really primed for future work.
So will you be doing this?
I don't know, not next year or this coming summer, but maybe in the future, trying to decide.
There's both mitochondria in the symbiont and in the host, correct?
Correct, yeah. But you know, the result did seem to show, since the aposymbiotic anemones didn't have a boost in superoxide production, I think it's localized to the symbiont.
Well, thank you, Caleb, for an excellent talk.
Thank you.
Our next speaker is Clare Zabel. The title of her talk is Linking Biogeochemistry and Benthic Community Composition at Palmyra Atoll's Western Terrace.
Hi everyone, thank you for coming. And today I'm going to talk to you about an interdisciplinary study I took on in the summer of 2012, where we went to the Palmyra Atoll and we tried to look at how the seawater chemistry was interacting with the community composition of the reef there. So I was advised by Professor Rob Dunbar and Kevin Arrigo, and other members of the lab gave me lots of great advice. So first of all, what were we doing? We went to the Palmyra Atoll. You would be excused for not knowing what that is because it's a very small and remote island with very few inhabitants. It has no permanent population, just a research station where scientists and Nature Conservancy staff stay and man the research station. It's a healthy reef, and that's one of the reasons why we chose it, is because it's one of the few areas where you can separate global forces that are affecting coral reefs, such as climate change and ocean acidification especially, from local factors that are also stressing reefs, like overfishing and habitat loss. So there's no habitat loss and there's no overfishing there because there's no one there at all. So that's why we chose such a remote location. Palmyra Atoll's reefs are known for having high biodiversity and high biomass at top trophic level, so lots of sharks and big fish. It's a really fun place to explore and a very healthy reef. And so when we were there, we were looking at coral calcification and the calcification rate, which is often used as a proxy for reef health. And so when corals are healthy, stony corals, they use their calcium carbonate skeleton to grow and become larger. When the ocean becomes too acidic, on the other hand, their skeleton starts to dissolve and they stop growing and can even start to die because of that. So, you know, people often use coral calcification as a way of trying to get at coral health. It's one measure, and I was trying to see how accurate of a measure it is when we look at these reefs that are suffering from ocean acidification and becoming more vulnerable to dissolution. Instead of calcifying, they start to dissolve. And so we estimated the calcification rates there from the changes in total alkalinity. So when corals are healthy and they're calcifying, the alkalinity drops. That's what we're hoping. And you can see that we chose several locations distributed around the western terrace of Palmyra Atoll. That's because that's a big open area where there's lots of corals for many hundreds of meters, pretty homogeneous throughout. And so we expected the alkalinity to drop when we looked at it during the day when corals were supposedly going to be healthily calcifying. And we wanted to make sure that the areas where we saw these seawater chemistry effects, this calcification, corresponded with actually healthy corals, because as the climate continues to change, we start to rely on these proxies. But if these proxies aren't accurate and we see that the seawater chemistry signal does not match up with corals that look healthy, that have high biodiversity, where there's young corals that are growing, then that's a problem, because we might be using a very inaccurate proxy for coral health, and we might not be focusing on the corals that really need our help most in the future. So we headed to Palmyra in the summer of 2012, and I was trying to answer this question: do biogeochemical markers of calcification and reef health actually align with observations of thriving coral communities? How can you do this? So we used a technique called Lagrangian drifts. And a Lagrangian drift, the basic idea is that you want to stay within a water mass. And as that water mass moves over the corals, you see changes as the coral's metabolism affects the water mass chemistry. So if you stayed in one spot, you wouldn't see the alkalinity drop because the current would just be moving new water in from the open ocean. But if you stay within the water mass as it moves over the surface of the reef, then hopefully you can start to see the effects of the reef metabolism starting from when the water first enters over the reef and going on until the water has been moving over the reef for a long period of time. And so we tried to do this by following one single water mass. And to do that, you can see we had our little drifter, which was very technologically advanced buoy, bucket, and CTD logger attached to it. And we kind of followed that guy around in a boat, hoping that it was following the water mass. And when that was happening, we would take water samples every 10 minutes. And we would also videotape the bottom cover at the sample site every 10 minutes so that we would have a set of ecological data that corresponded exactly with water that we could look at the seawater chemistry of later when we reached the lab. So that's what we did. Every 10 minutes, I would follow it around, the drifter around in the water with a camera that was on a pole taking pictures of the bottom. Then I took all of those images, and from each video at each sample site, I would grab 5 frames that were equally distributed in a circle around where the water sample was taken. And I divided each frame into a 10 by 10 grid. So that would be 100 small rectangles. And then inside each rectangle, I looked at what the bottom cover was to see if it was coral, if it was algae, if it was sand. And I looked to see what those were, and then hopefully the idea was that we could get a total percent cover at each site, and then add the 5 back together and get an average for each sample site. And so the bottom covers that were present with high frequency in the area of Palmyra we were studying were stony corals, halimeda, which is a type of calcifying green algae, corallimorphs, which are sort of like small anemones. They're cnidarians, but they don't undergo calcification and they don't have a hard skeleton. Turf algae, which is just a soft kind of green algae that doesn't have a lot of structure. Non-living material, which was almost completely ubiquitously white sand or dead coral. So all calcium carbonate material, and a few miscellaneous pieces that never amounted to more than a few percent. But those could be sea cucumbers or crustose coralline algae, something like that. So I added all of the data together, and in that way I was able to have a perfectly matched seawater chemistry set with an ecological data set. So what were the results? We analyzed the results in a few different dimensions. I'm going to talk to you about some of them, not all of them, because we looked at it from a lot of different perspectives. But the main thing that I was really interested in was seeing how well we could see that corresponding alkalinity drop with high coral cover. Because everyone is inferring that, or many studies are inferring that there's high coral cover or a healthy reef from alkalinity that's dropping rapidly. So is that really true? So here are the basic results of the most representative data sets that I found. On the top, you can see the alkalinity over the course of the drift. So it starts at the beginning of the drift and it moves chronologically towards the end of the drift. And then right beneath each set, you have a picture of what the bottom cover was like at each of those sites. So they match up as that moves, as the alkalinity changes on the y-axis, so did the coral cover and the other bottom covers. So you can see it's a real mix when you look at it, just sort of without any close examination. You do see some alkalinity dropping, but you also see some alkalinity rising, and most of them have very high coral covers. You might be surprised, some of you who research corals, to see such high coral covers. We purposefully chose areas that seemed to be to have very dense coral cover to start our drifts and then follow the water currents, because we wanted to see, since this was a new technique, we wanted to maximize the chance of getting a signal from the corals. So it's a very healthy area. The fact that there's some areas with upwards of 80% coral covers does match up with other studies of Palmyra Atoll, although it's a bit on the high side. And then there's a few that are lower, and on the bottom, I selected those. And then I just want to draw your attention to the two that had such extremely rapid alkalinity changes that I had to put them on a different scale. So you can notice those were both much, much steeper changes than the others were, one going down and one going up. And because they're also very smooth changes, it does seem like that might not be the result of two water masses mixing, but some sort of very rapid change. So that was interesting to me. And here I have a diagram depicting, to give you a sense of, if you started at the absolute beginning of where a water mass enters the Western Terrace, and then you just followed that water mass over the entire Western Terrace, what would the bottom cover look like over that period in time? So just to remind you, if you forgot from the last graph, the blue is stony coral cover. So as you can see, high coral cover, it does gradually start to decline as the depth gets greater and there's a little more wave action. So I was trying to figure out, why are these not corresponding quite as well as, or as closely as some people had thought that they might. So the first thing that jumped to my mind was time of day. Corals calcify more when during the day, but not always evenly. Sometimes if there's too much sunlight, they can start to decrease in calcification. If there's not enough sunlight, they also decrease in calcification. So on this graph, what you can see is that I have the total alkalinity of each drift, and then on the other axis, the time of day. So these were actually taken on different days, most of them. But if you just were trying to look at processes that might change depending on whether it's morning or afternoon or evening, this is what you would find. So what I thought was really interesting that jumped out to me is this area right here, where you have the two drifts where the change was very sharp, one of them declining and one of them increasing again, right around the same time. So it seems as if, for some reason, perhaps, calcification drops rapidly during that time of day, which was not a result anyone had seen before. And then the other thing I was wondering about was, because we sampled every 10 minutes and we sampled by time, not by distance, you could get an effect from water that was traveling more rapidly at some times than others. So if you have one water mass that's drifting much more quickly, it can cover more coral and potentially be more affected by the metabolic effects of the coral than a slower or than a faster moving one would have. So on this graph, I put water mass velocity with how much of a change you saw in the alkalinity. And as you can see, as the water goes faster, you do start to get more of an increase in the average alkalinity, but it's not such a strong effect, and there's a lot of noise there. So we also looked at a number of other factors that I unfortunately don't have time to get into, but we looked at effects from weather. We were curious about whether maybe there was some sort of strong storm or some clouds that potentially affected coral calcification during some of those drifts. And there wasn't any consistent pattern. There were light showers for a few of the sampling sites on a few of the days. Storms in Palmyra tend to pass very quickly. So there was no consistent effect there. We also looked at whether there could be an effect from accidentally moving into a different water mass. So maybe a different water mass moved in and we went into it and we didn't realize, and that's why you see a sharp change in alkalinity. So the way we tried to control for that was to look at, because we had the CTD logger, any really sharp change in salinity. When we saw a sharp change in salinity, we assumed that there had been water mass mixing and we discarded that drift. So all of the ones that remained were ones where there was really no sign of a sharp temperature or salinity change that would indicate water mass mixing. So we don't think it's water mass mixing. So my theory is that it's a combination of some of these weak effects that you're seeing. And I would be very curious to see more data on whether these corals are changing their calcification rates really rapidly around midday, like those two drifts hint that they might be. So those are the basic results, and I think that they have interesting implications for some of the reef management that we're looking at in the future. So first of all, it shows that clearly reef cover differences over pretty small spatial scales can have a big effect on measurements of total alkalinity. So if just a few 100 meters difference can indicate something very different about reef health, you have to be very wary of where you're taking measurements in the future if you're trying to use that as a proxy. And the other theory we had was that maybe we were wrong to just look at stony corals. Maybe it's some other sort of calcifying organism. There wasn't any really dominant other calcifying organism that we saw, although potentially the dissolution of some of the sand and dead coral could have also been affecting the seawater chemistry, which is something that, to my knowledge, no one has looked at. And I think this is the important one to me, which is that these really show that it's not easy to infer something about reef health from either just looking at coral cover percent or looking at alkalinity and calcification rates. Even together, you can still get some very mixed results. So that's a little bit frustrating because going into the future when we really need to monitor coral health, it seems like it might be a pretty complex task. And furthermore, this project, we tried to do it in a relatively low-tech way because we were looking for technology that can be used widely in the world to monitor reef health, not just in areas where they have really advanced research facilities, because as corals start to suffer from climate change and ocean acidification, it would be really helpful to find an easy way for reef managers in different places to monitor the health of their reefs. So we need ways to do this faster. Advances in automated image analysis would certainly be helpful because these images were analyzed by hand, which takes a lot of time. So that is something that we want to keep looking into in the future. I just want to thank some people who made this research possible. My amazing advisors, Rob Dunbar and Kevin Arrigo, Dr. Lita Teneva, who sadly has left us, but was a wonderful mentor to me when we did this. Tim Calver, you saw some of his wonderful photography. He works for the Nature Conservancy magazine and did an article on our research and kindly shared some of his pictures with me for the purpose of this presentation. The wonderful people in Earth Systems, of course. Amanda McNary, because she helped me with some of my figures, and all the generous funding that I got from different sources at Stanford and the Dunbar Lab got. Thank you very much.
We have time for a couple of questions for Claire.
Two quick questions. How did you measure alkalinity? Because it's quite difficult. And then also, how does changes in alkalinity from calcification compare to changes based on just like primary production altering?
That's a
So the
good.
questions are, how did you measure change in alkalinity, and how did alkalinity relate to primary production?
Yeah.
Other metrics.
Yeah, so we actually used, I forget the exact name of it, but it's the new machine that the Dunbar Lab actually got just that summer, and it was the newest one, so it should be the most up-to-date fine scale. We spent a long time calibrating it, and it was very sensitive to even very small changes in alkalinity. And primary productivity, to my knowledge, it's another way of kind of getting at what sort of photosynthesis the coral reef is undergoing. You need more data to get it reliably than the alkalinity, so we just didn't have it on as many of the Lagrangian drifts, but it would be interesting to use that too in the future.
Yes.
So just a quick follow-up on Caleb's question. You commented on how sensitive your measurement is. So you look like your toes were 10 to 15 feet above the coral surface. Do you need to be any closer to be able to get a stronger correlation between actual alkalinity due to calcification as opposed to any of these other factors that may be going on the reef?
So the question relates to how far away from the coral reef the measurements are being made.
So actually, that picture might have been a little bit misleading. When we took the actual water samples, that was me holding the camera. When we took the actual water samples, we had someone free dive down to the bottom of the reef and would take a sample 6 inches above the reef. It would be interesting to see, what the perfect amount above the reef would be. We chose 6 inches.
Yes, Katie.
So I have a question. You brought up a really good point about trying to develop methods that are using low-tech analysis. What would be your recommendations in terms of what you learned so far from this project for what folks could do to try and understand what the health of their reefs are?
So the question relates to developing low-tech methods for measuring reef health, and what would be the next step for advising reef managers?
So I think, in an ideal world, I know Gareth Williams, another person who's done research at Palmyra, has been looking at using satellite data, which seems like it would be, although it would have not as fine resolution, a great way to have a lot of data about how reefs were changing very quickly. I would say at least some sort of software that was easy to use that could analyze at a very crude level percent coral cover or something like that would be very useful. We did try several, and the problem was that most of them would reject images of quality that human beings could clearly differentiate coral from non-coral. So that kind of gets to the problem of a low-tech or low-cost measure. If you don't have a camera that is taking the most high-quality photos, those aren't working yet. I have faith that they'll get there in the future.
We have one last question.
There are fish that are camouflaged that can do various colors. If I go out to the reef terrace in Half Moon Bay, I can tell without looking in the water when I catch one of these kinds of fish, where the bottom is covered with kelp or white sand or whatever, just by the color pattern. Has anybody ever tried that?
You mean to look at the fish that were present and to try to infer something about the bottom cover?
That could change their color like a chameleon.
To infer something about the reef health, or to...
You could tell what the bottom of the reef is because the fish are going to camouflage themselves to what's there.
Yeah, that's an interesting question. I don't think anyone has, and you could probably get some sort of interesting data from a lot of fishermen. It probably wouldn't capture really rapid changes in the reef health, though, because if a reef declined over, the course of a few decades, the fish populations, maybe they would die off quickly, or maybe they would remain.
Yeah, they can die.
Yeah.
Some of these things can change kind of probably on a day or hour basis.
Oh, interesting. Yeah, I don't think anyone has looked at that, but that's a really cool idea.
Thanks. All right, thank you very much, Claire.
Thank you.
Next speaker is Hannah Druckenmiller, and the title of her talk is Fisheries and Food Security to 2030.
Hi guys, my name's Hannah, as Richard just mentioned, and I'll be presenting on my analysis of global fish markets to 2030. The goal of this project was to forecast supply and demand for fisheries products in order to answer the question, will the price of fish be driven out of the grasp of low-income populations by global population and income growth? Or can global aquaculture keep the prices of these commodities within their economic grasp? And so the point of doing this type of analysis is to see where investment, technology, and policy interventions ought to be targeted in the years to come. So I'll start my presentation with an introduction to the importance of fisheries to global food security, a review of the global status and trends of fish production and consumption, and also a little bit about what other researchers have found in this area. Then I'll walk you through the model I built to analyze this question and the major drivers of supply and demand I looked at. And finally, I'll talk about my results and some key recommendations that emerged from the analysis. So my main motivation for this research was the important role that fisheries play in global food security. It's estimated that about 3 billion people derive at least 20% of their animal protein intake from fish products, and this dependence is highest in the developing worlds. Additionally, fish provide a range of micronutrients often deficient in the diets of the world's poorest households, including essential vitamins, minerals, and fatty acids. Since 1970, per capita consumption of fish has actually doubled at a global level to 19 kilograms per capita in 2010. And although this is only a small portion of the total calories people consume per day, it's related to significant health benefits, including coronary heart disease, mental health and retardation, and growth and development. Additionally, small-scale fisheries play a very important role in food security in some of the poorest countries of Africa and Asia, where they not only support diverse and healthy diets, but actually provide a source of income and employment to millions of people. So as I mentioned, per capita fish supplies have increased significantly, but there's been considerable variation across countries and income groups. For example, China drove most of this growth where their per capita supplies increased from only 5 kilograms in 1980 to over 30 in 2010. Unfortunately, stagnant wild catches and rising feed costs for a number of major farm species are actually putting upward pressure on international fish prices, as you can see in the graph. And so this raises the question whether fisheries will be sufficient to provide affordable protein to the poor. Aquaculture has actually driven most of the growth in total fish supply since 1980. As you can see, production of farmed fish increased by over 12 times during this period. At the same time, capture fisheries production has remained relatively level since the mid-1990s at around 90 million tons per year. And capture fisheries, although total catch hasn't varied by very much, there's a big difference in fishing pressures across regions and species, leading to a wide range of exploitation statuses for marine ecosystems. So currently 30% of marine ecosystems are considered fully exploited, with another 50% overexploited. Sorry, that was 30% overexploited, 50% fully exploited. Okay, so here's a little bit about previous studies that have looked into international fish prices. You can see projections from the International Food Policy Research Institute, World Bank, and OECD FAO on the slide. And these results reflect that global demand will outpace global supply, leading to price increases across all commodities. The model I built is a supply and demand model that calculates world prices for fish products on an annual basis to 2030. And so how it works is you forecast production and consumption, and then you set regional demand equal to local production plus net trade, so either imports or exports, at a level that clears the world market. And so you're left with prices that equilibrate supply and demand. To simplify the model, I used 4 broad categories of seafood items: low-value food fish, high-value fin fish, crustaceans, and mollusks. And I forecast production and consumption in 11 geographic regions, which tend to follow traditional groupings, except for you'll see that China is singled out just because they have such a large share of global fish production and consumption. So this is the demand function that I built for this study. It includes a number of parameters specific to each regional market and to each year. The major drivers include global population and income growth, as well as the effects of changing tastes and urbanization, which tend to increase demand for high-priced calories. Additionally, demand responds to changes in price through elasticities of demand, and it also is set on a yearly basis such that consumers have the opportunity to alter their decisions in response to changing markets. I forecast supply separately for aquaculture and capture fisheries. In the top function, you'll see aquaculture, which was driven primarily by factors such as investment, natural resource availability and constraints, supply chain infrastructure, and regulatory regimes in major producing regions. And then supply is down below, and that's influenced more heavily by historical trends in production, as well as stock assessments. And I know that this is not making sense right now, but I'm going to dive into each of the main drivers in order to give you an idea of how I assigned values to each of these parameters. So on the demand side, the main drivers are population growth, economic development, and price effects. You can see in the graph that the global population is expected to increase from about 6.9 billion in 2010 to 8.3 billion in 2030. And most of the growth is expected to be concentrated in the developing world, where fertility rates exceed replacement level, and a disproportionately large share of the population is within or entering their reproductive years. Income growth is another key determinant of fish demand. I got these projections from the Total Economy database. And basically what's happening is that GDP growth rates are declining in most of the world as economies become wealthier. However, there is regional variation. For example, in recent years, growth rates in Latin America, South Asia, and Sub-Saharan Africa have increased steadily. So projections for these regions range between about 4% and 8% per year. On the other hand, in developed regions, projections range from about 0 to 3% per year. So on the slide, you'll see per capita GDP projections. These were then translated into changes in quantity demanded through income elasticities. And so elasticities are a tool which translates a change in either price or income into a change in quantity demanded. The values I used are projected on the slide. because fish is a luxury good, income elasticities and own price elasticities fall between about 0.5 and 1.5. And so you'll see that income elasticities are positive. And this makes sense because if your income increases, you can afford more fish. On the other hand, own price elasticities are negative, reflecting the fact that if the price of a good increases, your consumption of that good is going to decrease. Cross-price elasticities describe the change in the quantity of fish you demand in response to the change in the price of substitutable goods. So those are positive, and they tend to be smaller than the other two. Okay, now I'd like to dive into the drivers of supply, starting with aquaculture. As I mentioned, it's been aquaculture that has driven most growth in total fish supplies over the last 20 years. From 1980 to 2010, the average annual growth rate of the industry was almost 9%, making it the fastest growing food sector in the world. However, this rate appears to be slowing, as over the last decade, the average annual growth rate was only 6%. Within the industry, there's a strong trend towards intensification of production systems in response to growing natural resource constraints, such as freshwater availability, coastal zone pressures, energy requirements, and limited feed supplies and price levels. So recognizing that improving resource use efficiency in aquaculture is already a priority within the industry, there are two main approaches. The first is improving management, and the second is the development of technological solutions. Currently, less than 10% of aquaculture depends on genetically improved stocks. So that means there's a lot of room for improvement through selective breeding and also transgenic technology programs. Additionally, systems engineering approaches have a high potential for impact. So an example would be recirculating aquaculture systems or offshore cage technology, which really reduce or optimize your water use. Additionally, integration approaches are really growing in importance. Many of you may be familiar with integrated multi-trophic aquaculture, which is one example. And so that's when a farmer can use the waste stream from one production system as the inputs to a second production system. And lastly, just like demand is affected by changes in price, so is aquaculture supply. So taking all these effects into consideration, you can see the projected growth rates for aquaculture over the next 20 years. They start out between about 3 and 6% in 2010 and then decline to between 1 and 3% in 2030. I approached forecasting supply for capture fisheries a bit differently just because it depends on different factors. So the main things I considered here were historic trends in production, stock assessments, environmental changes, and then once again, price effects. As I mentioned, total global catch hasn't varied much since the mid-1990s. It's actually been by less than 3%. But there has been significant variation in regional catch and in catch among major species. So you can see on the slide the 3 broad temporal patterns that we can characterize these trends into. Increasing, like in Southeast Asia, decreasing, like in Central and Eastern Asia, or large oscillations, like in Latin America. Additionally, stock assessments played an important role. I looked at stock assessments for each species in each region, and the global numbers are given there once again. In the coming years, environmental stressors are also predicted to have an increasing effect. So these might be things like climate change, ocean acidification, nutrient runoff, and industrial pollution. For example, recent research suggests that climate change is actually going to increase catch potential in high latitude regions, but decrease it in the tropics. And lastly, price effects were a major driver of supply. Interestingly, it's estimated that price only influences about 12% of capture production, which is much less than the 99% of aquaculture production it influences. And that's just because regulatory and resource constraints in marine capture fisheries really lead to price inelastic supply. So once again, you can see the growth projections for capture fishery. These ones range from about 0 to 1.8% per year, and they have large variations across regions and commodity groups. Any discussion of global fish markets would be incomplete without a discussion of fish meal and fish oil. Approximately a quarter of the wild-caught fish are not consumed directly by humans, but rather reduced into fish meal and fish oil for use within animal and aquaculture feeds. And production of these commodities has been pretty stable for the last decade, between 5 and 6 million tons per year. But at the same time, we've seen huge increases in aquaculture's demand for the commodities, which has led to a dramatic shift in the end use of fish meal and fish oil. So on the slide, you'll see that in 1960, only 2% of fish meal and fish oil went to aquaculture, and in 2010, 73% did. And these gains came largely at the expense of pig and poultry feeds. But the problem is that aquaculture's demand keeps growing and the supply is stagnant. And there's actually no commercially viable substitute for fish meal and fish oil within aquaculture feeds because we don't have a commercially viable source of long-chain omega-3 fatty acids. So there's this really urgent need for substitution away from these commodities. Fortunately, inclusion rates in feeds have been declining for decades. On the slide, you'll see the composition of Norwegian Atlantic salmon diets from 1995 to 2010. And just over this 15-year period, you can see large declines in fish meal in blue and then fish oil in gray, with increased use of plant and animal-based substitutes in the green and the purple. However, further gains are still necessary. Some solutions are farming fish lower on the food chain or expanding the use of animal and plant-based substitutes. We could also produce single-cell oils to make fatty acids or genetic engineering of oil seeds to produce these long-chain fatty acids. And then the last one is using fisheries byproducts or trimmings as a source of fish meal and fish oil rather than wild-caught fish. So putting all these major drivers of supply and demand together, I came out with a set of projections to 2030. I'll present first my baseline projections, which represent the most likely scenario based on current trends in production and consumption, and then I'll briefly talk about the sensitivity analysis I conducted. You can see on the side the projections for fish supply to 2030, which increased by over 50%. Most of the growth is driven by aquaculture, which accounts for 86% of the increase in total fish supplies. Here are the consumption projections. You can see that at a global level, consumption increases at a rate about 1.6% per year, and the fastest growth rates are in sub-Saharan Africa and South Asia, hovering between 5 and 6%. At the same time, we're actually seeing declines in per capita consumption in some of the more developed regions, like North America, Western Europe, and Oceania. On the slide, you'll see the real price projections for each commodity group. These are actually pretty interesting because the prices decline in the first 5 years of the projection from 2010 to 2015 for all commodity groups, led by low-value food fish. And we see these large declines in price due to high rates of aquaculture development and then also increases in inland fisheries landings across Asia. Then starting in 2015, the prices actually all increase. And this is due to high levels of economic development and population growth in the developing worlds. In the end, you see mollusks increase by 16% at the bottom end, and then at the top end, low-value food fish actually increase by 26%. In addition to my baseline scenario, I looked at 12 alternative scenarios. I don't have time to go into all of them now, but I'd like to just touch on a couple points. First of all, these scenarios were conducted in order to illustrate how sensitive the model is to changes in a number of key parameters. So for example, higher capture landings or lower aquaculture development. And the model was most sensitive to changes in income elasticities or alternative fish meal and fish oil assumptions. It was also altering the productivity assumptions of capture fisheries had only half the effect of altering the productivity assumptions of aquaculture. So given these results, I'd like to conclude with a number of key recommendations. The first thing you'll notice is that aquaculture continued to drive total fish supplies, but the question remains what social and environmental costs this development will incur. Price signals could provide a strong incentive for technological change within the industry, but the problem is that a lot of aquaculture's major costs, like environmental degradation, aren't actually priced in the market. And so this implies that we might need some policy interventions. Governments in major producing regions ought to establish enforceable standards that set clear limits on natural resource use. Another thing you'll notice is that capture fisheries production remains relatively level. It's my hope that global fisheries can be rebuilt by the implementation of new management regimes. One promising approach includes combining traditional management approaches like catch quotas with economic incentives and the targeted placement of marine protective areas. And then a last result that stands out is the large price increases expected for fish meal and fish oil, which were about 25% and 50% respectively. And so these, again, highlight the critical need for continued substitution away from these commodities. In terms of food security, per capita fish supplies are increasing, but at the same time, the prices of all these commodities is increasing, which implies that there will be a less equitable distribution of fish products across income levels. Additionally, we're seeing a lot of regional variation in where fish supplies are increasing. Sub-Saharan Africa and South Asia, as I mentioned, enjoyed the rapidest gains in per capita consumption, but these two regions were also projected to become heavily dependent on imports, which might expose them to greater price volatility and vulnerability in terms of food security. And then lastly, given the scenario and the baseline results, I think that the study highlights the importance of continued investment in aquaculture. And so some approaches might be to invest in education and R&D. Governments should work on ensuring the availability of affordable feed and seed and improving access to capital and markets for fish farmers. And lastly, it's important to promote both a mix of small and large-scale farming operations so that you can provide an income-generating opportunity to the poor, as well as support larger macroeconomic growth and gains in food security. Lastly, this model did require a considerable degree of simplification, and talking to you about it today required another degree. So there are definitely improvements that could be made if I included data at a finer scale, such as countries or species-specific data, and that would increase the resolution. But it's my hope that this exercise acted to illustrate the key dimensions of global fish markets and how they're evolving in response to technological, environmental, socioeconomic, and demographic changes. Thank you guys all for listening, and a big thanks to my advisor, Roz Naylor, who's been just an incredible mentor over the last couple of years.
We have time for one question. Norm?
Fish farming can be incredibly destructive on the capture and catch. Like the introduction of silver carp and snakeheads and stuff into the United States. And did you take that into account any that the fish farming will be elsewise destructive and its gains will be offset?
So the question is did you think about or consider the environmentally destructive aspects of fish farming?
Yeah, so aquaculture is actually a really interesting area because at first people thought that it would add to global fish supplies because farming should take pressure off some of capture fisheries. But because you require wild feed inputs to farm fish, and because it takes up habitats along coastal zones, introduces exotic species, and introduces disease, it's much more unclear at this point. Roz, who's my advisor, did a study a couple years ago that showed that kind of the amount of fish you put in and get out is increasing, so that aquaculture now is a net contributor to global fish supplies, but there are an incredible amount of environmental costs. And if with an industry growing this quickly, it's really a priority to make sure that the effects, industrial agriculture has had all these severe environmental and socioeconomic effects, and so it's our chance right now to try and stop that with aquaculture.
Thank you very much. To remind you that at the conclusion of our symposium today, around 5:20 p.m., Madison will be toasting all of the presenters today and their mentors. So if you have to leave after the session, I encourage you to come back and enjoy a glass of champagne with us a little later this afternoon. So as I mentioned, Clare LeDuff is our next speaker. She's got a beautiful photograph of Speyeria mommonia, the Mormon fritillary. If you've had a chance to visit some of the local mountain ranges in California or the American West, you certainly come upon this beautiful butterfly. So Claire's working on exploring the potential effects of climate change on protandry in the Mormon fritillary.
Hello. So as Richard said, I'm looking at the potential effects of environmental factors on protandry, which is a reproductive strategy in this study species, Speyeria mormonia. So first I'm going to give a little bit of background, general picture of my research, and then I'll describe my study organism and explain what protandry is, and then I'll delve into my research question and methods and finish up with results and discussion. So the most general version of my research question is, how are insects affected by climate change? And insects tend to be affected, or are thought to be more affected than other organisms because their life history is so tied to the seasonality of their environment. In addition to the fact that that their high reproductive rates and quick generational turnover means that environmental adaptations show up more rapidly than in longer-lived organisms. And so we're actually already seeing the effects of climate change on insects worldwide through range shifts and shifts in community composition and interactions, and most importantly for my question, in changing phenology. And phenology is what I just mentioned. It's the seasonality of their life cycle. So I did my field work this last summer in Gothic, Colorado. I was a research assistant for Professor Carol Boggs, and she was a professor here at Stanford up until this past year. She's working in the bio and hum bio departments, and now she is the director of the School of the Earth, Ocean, and Environment in the University of South Carolina. So Professor Boggs has been collecting population data on this species of butterfly since the late 1970s. And her study site is right outside Rocky Mountain Biological Laboratory, or Rumble, which is in the Rocky Mountains in Colorado. And she focuses on the population dynamics and resource use of the species. But I'm going to talk about the sexual reproductive strategy of the species. So Spiraea mormonia lives in, as Richard said, the western United States and Canada. And it lives in flowery alpine meadows, like the study site that's right outside of Gothic. And yeah, so this is its life cycle. It starts, well, I'll start at the top. I don't really know where the beginning is, but so the eggs hatch in the late summer, and larvae or caterpillars diapause during the winter, and diapause is a stage of suspended growth, like hibernation kind of, so that they can survive the harsh weather conditions of the winter. And instar refers to the phase between molts. So molting is how larvae grow. So first instar means before they've molted, so they go right into diapause after hatching without feeding. And then come spring, the larvae pupate, which means that the caterpillars form a chrysalis, and they'll emerge in the summer. The males emerge in the early summer, and females emerge a few days to a few weeks afterwards. And during the summer flight season, which lasts from June to late August, they will mate, and females lay their eggs. They lay them singly on leaf litter that's around violet plants, which is the larval host plant, which means the plant that the larvae will eat when they hatch. And by the end of the season, all of the adults have died off, and the new generation will hatch, and we're back at the beginning. So what is protandry? Generally speaking, protandry is when the males of a species arrive before the females. And so protandry is seen in mammals, fish, reptiles, amphibians, birds, and insects. And so in birds, for example, it can refer to when the males physically fly to the breeding location before the females. But in this butterfly, it refers to when the males eclose or emerge from their pupa earlier than the females. And so what is the function of protandry? There are a lot of very complicated models to explain the function of protandry, so I'm just going to explain the most accepted and the simplest version of it. So for butterflies, it's thought that it's a beneficial strategy for both males and females, because all butterflies, after they emerge, need some time for their wings to dry and fully extend for them to fly. And males require a few days to sexually mature after they emerge, so they have some time before the females arrive, and so they're flying and sexually mature when the females emerge. And in addition, Spiraea mormonia females generally only mate once, so males want to encounter unmated females, so the earlier males are more likely to encounter a female as she's emerging and before she's been mated. And it also benefits the females because it decreases the amount of time they spend in the adult stage unmated. So they're more likely to reproduce before they die. So my research question is, do summer weather conditions affect the degree of protandry in Spiraea mormonia? So do weather conditions affect the amount of time between male and female emergence? And we know from past research that male emergence time is very tied to snowmelt, and it's true of butterflies that the ambient temperature affects when they will emerge. So I was curious to see if, particularly with climate change and with the changing environment, if factors will affect male and female emergence differently and therefore affect the degree of protandry. So I defined the degree of protandry as the difference between the female peak flight date and male peak flight date. And so this is just an easier proxy for a difference between the emergence times. And the environmental factors that I chose were average summer month temperatures for May, June, and July. And these are the months when the adults are going to be emerging and flying. And then lastly, also snowmelt date, because I know that we know from past research it affects particularly this population. So we have two main hypotheses. The first of which is that these environmental factors that I chose are in fact changing over the study period, which is 1982 to 2012, and I'll explain why that is in a minute. And so I hypothesized that snowmelt is in fact moving earlier in the season, and that these average monthly temperatures are increasing. And my second hypothesis is that protandry is affected by an interaction of these factors. So, like I said, male emergence is tied to snowmelt date, so I expect that with earlier snowmelt date, males will emerge sooner in the season, which would increase the degree of protandry. But with hotter July temperatures, it's likely that female development will speed up, and that would decrease the degree of protandry seen. So I'm exploring the interaction of these forces. So to collect population data, Carol Boggs and her lab use mark-release recapture methods, it's a common ecological field method used to estimate the population size of an organism on a given day. So this is one of the research assistants I worked with in our study site, and we went out about every third day during the summer. It's pretty convenient because butterflies only like nice weather, so if the weather was bad, we didn't have to go out. And we would go out there, and our site was broken up into 6 subsites, and we spent 1 person hour in each site. So with 3 of us, we'd spend 20 minutes catching in a site. And after the 20 minutes were up, we would sit down and look at each individual and mark down if it had already been captured, because we mark each butterfly with a number. And if it hadn't been captured, we would give it a number. And so we would record its number, its sex, the subsite it was caught in, and wing wear, which is used as a proxy for age. And after that, we would let the individuals back into their subsite. So what do we do with this data? So we have counts of male and females from each MRR day, and using the Jolly index, which takes into account the number of new captures, recaptures, and individuals known to be alive on a given day because they're captured later in the season, to make a population estimate for every day of mark release recapture. And then once these are graphed, like this 2011 emergence curve example, we can interpolate between the MRR days to estimate the population on any given day throughout the summer. So we used these to calculate the degree of protandry. You can see the male emergence curve in green peaking before the females, and so male peak flight date was August 11th. And
that's the number of new adults? That's the number of adults hatched, or the total number of adults?
This is the number caught on a given day, so this is the population of a given day.
Okay.
Okay, sorry if that wasn't clear. Yeah, so this is the data that was taken from our MRR, from mark release recapture, and then using the Jolly index to estimate the population size on a given day. And then from there, we used peak flight date as a proxy for emergence time. And we'll peak the difference in their peak flight dates as the difference in their emergence times. So Carol has a large set of data that spans back into the 1970s. And I went through most of all the years that had mark release recapture data and chose the ones that had adequate numbers to make Jolly estimates of populations, and that ended up being 6 years between 1982 and 2012. And so that's why that's the study period. And then, like I said, I graphed these and found the degree of protandry for each summer. And from there, I tested linear regressions of protandry and the environmental factors that I mentioned. So this is my final data set. You can see the protandry, each of their peak flight dates, and the summer weather conditions that I tested.
Is there a bias, because you're biased towards the years the butterflies are most abundant? There's no butterflies around.
Let's just wait till we
go ahead, yeah. Okay, but it's got me on, yeah. Good.
Okay, so my results. The first hypothesis, I found that 2 of the environmental factors I was testing were responding as I predicted. So snowmelt date is moving earlier in the year over this time span, and July temperatures are increasing. And then for the second hypothesis, I found, as expected, that the male peak date has a significant relationship with the snowmelt date. And then a trend that I didn't expect was that the degree of protandry is decreasing over time during the study period. So what does this all mean? So in the end, I didn't end up finding any significant relationships directly between these environmental factors and the degree of protandry, but I did find some other interesting things. Like I said, the climate factors are changing with the summer temperatures warming and snowmelt occurring earlier in the season. And so these might not directly affect protandry in this species, but definitely have the potential to indirectly affect spirea through resource availability. So luckily some research has already been done at Rumble, where the study site is, on the nectar sources for spirea, and found that early snowmelt causes a bimodal distribution of flower abundance during the summer. So instead of a unimodal one that matches the flight, the emergence curve of the spirea in a summer, there's a bimodal one that leaves a trough of low flower abundance in the middle of the summer, which means that there would be less nectar per capita. And in general, earlier snowmelt also means like overall lower floral abundance. So male peak flight date is affected by snowmelt, but female is not, and yet the degree of protandry isn't changing. So something is affecting the emergent state of females. And if I have a larger sample size, then I could see that snowmelt date or the July temperatures is what is affecting it, but or it could be some other variable that I haven't looked into yet. And so if this means that the degree of protandry isn't changing, but male emergence is moving earlier in the season, then that means that the flight period itself is actually shifting. And though their sexual reproductive strategy isn't being affected, this again could have implications for resource availability or survivalship of the larva. So if eggs are laid earlier in the season, then those larvae that hatch have to survive for a longer period of time before they enter diapause. So like I said, there are definitely other variables influencing the results I found, and it requires more research. So I'd love to look into it with a larger sample size and maybe consider other variables like microclimate and day-to-day weather conditions, which also affect whether or not an adult butterfly will emerge on a given day. And the larger scale environmental factors that I took into account just because it's easier and makes more sense for like a long-term scale project, don't really take into account those short-term factors. So then there's also this pattern of decreasing protandry over time that's unexplained, and more research needs to be done to figure out why that's happening, and also the mechanism of protandry within this specific population needs to be better understood to know how that will actually affect this population. But in the end, I didn't find any significant relationships between these environmental factors and protandry, but it's clear that climate change is affecting, or these climatic factors are affecting the phenology of this species and the resource availability. So climate change will indirectly have effects on Spirea mormonia going into the future. And I'd just like to thank my advisor and first reader, Carol Boggs, and my second reader, Rodolfo Dierzo, and the Bio Department for my VPUE grant, and then the Earth Systems Department for their support.
All right, we have time for a few questions. Yes, sir.
Maybe you said this and I missed it, but was there a relationship between the protandry and the actual population size that you were estimating? Like, did the decreasing protandry correlate or cause like a smaller population?
That's actually.
So maybe repeat the question. So the question is there a relationship between population size and protandry?
That's actually something that I didn't really have time to go into in here, but I also I didn't look at population size, but I looked at sex ratio and how the sex ratio, the protandry affects sex ratio, which would indirectly affect the population size of the next summer. So it's something that is talked about a little bit in my thesis, but I didn't have time to go into here.
That kind of semi-answers my question. I think I may have slightly misunderstood your slide area, where you're only showing certain years.
Mm-hmm.
But if you could only get statistics on the years the butterflies are abundant, that kind of biases your observations to when there's large rather than small population size.
Yeah, that's definitely a factor in it, but it's also just there isn't MRR data from every summer. And some of the summers, like MRR data in itself isn't great because you go out and if the weather isn't great, you're not going to see as many butterflies as there really are. So on all ends, it's kind of just an estimate. So I understand what you're saying, that the larger population size makes it a viable year, but that's kind of all I had to work with.
You have to work with the data.
Yeah, yeah.
Are there any additional questions? Claire, I have a question for you. So you say that butterflies don't like the bad weather.
Mm-hmm.
And so that's part of what you're going to be looking at next. That was sort of the initial question, was what's the connection between weather and protandry? So what other...
I guess when I was saying that, I mean like day-to-day weather conditions. So because butterflies rely on solar energy to fly, to raise their internal temperature, that if the sun goes behind a cloud for 10 minutes, there won't be any butterflies, and then the sun will come out and there will be a ton. So that's another thing that I do want to consider, the day-to-day and like microclimate factors on the degree of protandry. But I guess I was just looking at more like longer-term averages of temperatures. Did that answer your question?
Oh, absolutely.
Okay.
Are they found over a range of elevations?
Mm-hmm. Yeah, so this is at like 9,000 feet, but...
So they should come out earlier at 5,000 feet.
Probably.
If they live there.
Yeah, I don't know. I just studied this population, so.
Okay.
All right, that concludes session 1. To our second oral session. Our first speaker is Zixiang Zhang, who is a graduating senior in geological and environmental sciences. And the title of his talk is Life History Dynamics of the Tiger Salamander, Ambystoma tigrinum in Response to Late Pleistocene Climate.
Thanks, Richard. Hi guys, my name is Zixiang. I'm senior in geology, and today I'll be talking about my thesis project, working on a fossil site in high elevation Colorado in the Eastern Rockies. And I'll be looking at tiger salamanders and using a fossil population of tiger salamanders to help us understand a global climate transition at the end of the late Pleistocene. Okay, so before we get started, I would just like to acknowledge some folks. First of all, I want to acknowledge my funding sources, both Stanford VPUE as well as the Stanford STEM Fellowship Program, in addition to funding from the Denver Museum of Nature and Science. I also like to acknowledge members of my lab group, my professor Elizabeth Hatley, as well as graduate students in my lab and interns for their help and suggestions. Folks at the Denver Museum of Nature and Science, especially Richard Stuckey, who oversaw my work at the Denver Museum last summer, as well as Jeff Pagatti, a geologist at the USGS, who actually helped to put together a lot of the detailed geologic history of the region that is extremely important for the context of my study. Okay, so first I would like to start off with just a broad overarching question on what kinds of questions I'm trying to look at. So the one big question that I have is, what do population level changes in tiger salamander body size and morphology tell us about changing environmental conditions across global climate transitions at the end of the late Pleistocene? So I'll go into detail in the next few slides about the tiger salamanders, but essentially we're working with a fossil record. So the first step in our plan is really to select some sort of osteological proxy or proxies, because all we have are the bones, and we're trying to use this massive collection of bones to tell us something about what happened in this area during the late Pleistocene. So we select some osteological proxies for life history. We then use these proxies to characterize some phenotypic and quantifiable traits of the tiger salamander population. So in my study, I've chosen body size and morphology simply because amphibians are extremely sensitive. They display many different types of morphological characters. Then what I do is I look at the sort of the population level patterns in these traits, and then I analyze the dynamics of these life history characteristics. So life history collectively refers to these phenotypically expressed traits of an individual that inform us something about the lifestyle of this organism. And finally, I want to use this population fossil record to reconstruct and constrain some of the paleo-environmental conditions and changes across this global transition. So just briefly talking about the Ambystoma tigrinum, tiger salamanders are endemic to North America. They are one of the most abundant families, genuses, of salamanders in America. And as you can see, they exist in a relatively unbroken range consisting of isolated populations throughout the continental US, from the Atlantic coastal plains all the way to the eastern edge of the basin and range boundary. And there's a lot of classification schemes for the tiger salamanders because they're such a complicated population and there's so much interbreeding going on that it's really hard to differentiate between subspecies, species, and even up to the genus level. For the purpose of my study, I'm examining a population using this classification scheme, which is one of the more popular ones, that has 7 subspecies. And I'll actually be looking at the Arizona tiger salamanders, Ambystoma tigrinum nebulosum. And just to talk about some of the existing records that we have of the tiger salamanders, there's actually not much out there in terms of fossil populations. I mean, there's a lot of studies of modern populations in the Rocky Mountains, but in terms of fossil populations, this is all we have. We have some studies from the 1940s and 50s, one from the 1980s, and one in 2005 that was done in my lab with the Hatley group. There's also some scattered research in Arizona, but there's barely any fossil record, and a record that consists of 80,000 years of record, which is what we have, is something that's extremely unique to our record. So our site is here in East Central Colorado. And this is a better look at our study site. So we are looking at a paleo lake basin, essentially a basin that used to be a lake formed or bounded by glacial moraines that is now no longer a lake. And it's located right here, East Central Colorado, in the Elk Mountains. And the lake formed at the end of the Bull Lake glaciation about 130,000 years ago. And the entire fossil record spans 130 to 45,000 years ago. And this is a really interesting period because what we have here is we have the Bull Lake glaciation at the beginning of our record, and then at the end of our record, we actually have the Pinedale glaciation, which is a localized glaciation that's part of the Wisconsin glaciation. And between those two periods, we have the Sangamon interglacial. So the Sangamon interglacial is the last interglacial stage in Earth's history. And in terms of climate, it's actually very similar to our modern day conditions. So what this record allows is very close sort of comparisons between a fossil record and a modern record that at the same time allows us to apply modern understandings to better understand the fossil record that occurs on a much longer time scale. This is a high elevation site above 27,000 meters. So it's above the snow line, and that's a type of fossil record we rarely ever find in the world. In fact, this site has been touted as the best high elevation fossil excavation site in the world in terms of quality and preservation. Finally, this is an isolated lake basin, which makes it really interesting because we're studying one single population, which makes it that much more powerful as a proxy because this entire population captures changes in one area over this amount of time. Okay, so some pictures to go through. This is what the basin would have looked like 130,000 years ago with the retreat of the glacier and the formation of these recessional moraines that are bounding this site, this high elevation site, that's allowing sediment to eventually accumulate in the basin, and that's allowing for moisture to subsequently accumulate in this basin. So this is an image of what the site might have looked like, sometime in the Sangamon interglacial, let's say 120 to about 70, 80,000 years ago. Really lush. This is most likely summer as there is no snow, and there's a lot of pines and spruces and large megafauna, but that's not what we're interested in. So this is what the site looks like nowadays. It's on privately owned land, a Ziegler Reservoir, and all the sediments that contain fossils have been excavated, and the excavation started in 2010, and now the site has actually been dammed and closed off to the public, so the site does not exist anymore. Okay, so this is a stratigraphy of the site, basically consisting of, it's very classic lacustrine facies containing the sort of inner fingering of lake sediments, so mostly silt, predominantly silk lake sediments interfingered with periodic debris flows that capture sort of snapshots in time. And these are sediments that have been moved from land into the water in these broad sort of wash deposits. And our tiger salamander fossils have been sort of gathered from the lakeshore sediments, whereas most of the high profiles, fossils, the mammoths and the mastodons are coming from the lake center. So relatively speaking, this area is not as well studied. And this is a look at some of the elements that we find in our collection. So I have in my lab boxes and boxes of these bones, all isolated and unidentified. So my job initially was just go through 25,000 individual bones consisting of these elements, and then and basically identifying specimens and pulling out interesting specimens. And what I'll be talking about today and for the rest of this presentation is the vertebral column, which, as you'll see, will tell us a lot about the tiger salamander. So briefly, tiger salamanders are extremely complicated. There's a lot of modern studies on sort of their sensitivity to climate and habitat conditions. But what I want you to take away is that tiger salamanders, they provide an ecological proxy because they're so sensitive to the environment. So I just want to go through some, just this schematic that allows me to explain some of these changes. So these symbols represent wet, dry, cold, and hot. Of course, this is very simplified, but it gives you a general idea. To have these larval forms, especially the neotenics, you have to have permanent water. You have to have water that isn't seasonal. Therefore, when you have these neotenic forms, you can't have a marsh setting. You can't have something that dries up because these live in water their entire lifespan. When you have really dry conditions, that's when you get a high abundance of metamorphic forms, especially when you have dire environmental conditions. You get these young metamorphs, essentially larvae that metamorphose extremely early on. And just to look at correlations between temperature and size. So again, it's much more complicated, but what we can imagine is that we know that metabolic growth is slowed in colder conditions. So when you have a colder environment, what happens is the larvae actually grow for more than the time that they would usually grow because they're growing much slower, and as a result, they overwinter, which means they go through the winter without metamorphosing, and that allows them to grow to really big sizes. On land, because metabolic rate is correlated with temperature, you have the growth of metamorphs to extremely large sizes in hot temperature conditions. So warm temperatures, you have a large range in size. You have abundance of young metamorphs. In cool temperatures, you have, in contrast, large larvae that have been growing for a long time in the pond and not metamorphosing. In deep permanent water, you have high neotenic abundance. And finally, when you have overcrowding in a deep permanent body of water, you get cannibalistic morphologies. You get literally these salamanders that have been sort of osteologically distorted and enlarged in forms such that they can swallow their peers. What I've done is I've gone in and looked at, and using the tiger salamander vertebra, I've looked at some morphological traits, including age and habitat, but more so morphology. So I'm using the fusion of the neural arch to form the neural spine. I'm using that as an indication of maturity, of age. So I use that as a way to separate my juvenile from my adult classes. Then what I do is I look at the ossification of the centrum. So the centrum itself in terrestrial animals is normally ossified, which, and the stability of an ossified centrum of the centrum, right, allows movement on land. So because stability is something you need in water, but not really on land. So the ossification of the centrum tells us that this salamander has moved onto land and is terrestrial. So I use the combination of these two traits, as well as indications for cannibalism, which I just use a sort of randomly, not randomly chosen, but I just chose a size boundary above which I designate a specimen to be a cannibalistic specimen. So these are some of the traits that I'm using. I'm measuring the centrum width and centrum length and using those as proxies for body size. And finally, I just do a bulk count of all of my specimens, including the number of total identified specimens, as well as the minimum number of individuals from my collection. This is a picture of what some of the work looks like, just boxes and boxes of my specimens in little vial bottles. I look at them under the microscope, I photograph them, I identify the specimens, and I've stood in this spot for, I don't know, hundreds of hours? I don't know. But okay, anyways, so this is some of my raw data. These are the lithologic units. So just look at the last one. The P stands for pebble, the S for silt, the F for debris flows, and there's also a peat layer. So most of our specimens are pretty evenly spread throughout our units. The upper silt, I suspect, this was sort of an error in terms of stratigraphic designation in that the upper silt belongs with the beach silt, but for our purposes, I'm just going to leave it here. But most of our units are well represented, and if you look at the minimum number of individuals in our units, we have good representation. Most of our specimens come from our main silt unit and also our beach silt unit. We have data from the tusk, which is really interesting. We have this one tusk that contains tiger salamander fossil elements within the tusk. Unfortunately, we have not been able to date the tusk, so we don't know where it's from, which, I mean, it kind of loses a lot of its power. But what we do know is that this tusk actually contains a lot of metamorphic forms, if you look at the numbers. And this tusk essentially captures a snapshot of a very, very nicely provided for terrestrial environment in which all of these metamorphs are just hanging out in it. Okay, so now I'll go on to sort of just looking at some of my data in terms of sampling. These are not super important individually, but the important thing to get from this is that there is really good size distribution. We have really good sampling of our data, and this has been by lithology, which is also time. Now we bin our body size data. Oh, so I just want to mention on the X is our centrum length, which is the proxy that I'm using for body size. So if you look at the salamander vertebral column, a salamander has about, let's say 35 to 45 vertebrae. And that is actually linearly proportional to the snout ventral length, which is the length of the specimen. So we use centrum length as proxy for body size. Good normal distribution. That's something you also see when you bin the sampling by morphological class. So from this, we can more or less, we've also done some other tests for taphonomy. We've been able to remove taphonomy as a prime driver for any of our trends because we have such nice distribution of data. This is a look at the relative morphological abundance of our different morphological classes over time. So on the X, we have relative abundance, 0 to 1, so basically 0 to 100%. And also time, oldest on the bottom, 140 upwards to about 55. So what this shows is that we have consistently low juvenile abundance, which is not surprising. What is interesting is that this is a pollen diagram. Our pollen diagram captures basically two sort of transitions in terms of relative abundance. First, the first one right at the beginning of the interval in which we have a decrease in metamorphic abundance and an increase in neotenic abundance. And then another sort of transition in which we have a decrease in the metamorphic abundance and an increase in the neotenic abundance. And these two patterns are things that I will go into in a bit. I'll summarize them. Okay, this is another look at our data, and what the takeaway point of this diagram is that we can actually use centrum length and centrum width, which are two very simple sort of quantifiable traits of the vertebral column, to tell us something about growth rates. So in this, for the purpose of this analysis, I use centrum length as a proxy for the length of the body, SVL, and then I use centrum width to represent tail height, the height of the tail. And this is interesting in the sense that after using Lewis curves, which are locally weighed sort of scatter plotting, in which you basically take into account local variations in the scatter and plot a line. And I've been able to, after looking at this, I've been able to say that essentially what you have is in metamorphosed forms, you have a slightly higher ratio of growth in body length versus tail height. And what this is telling us, and this makes sense when you look at a tiger salamander in real life, in the sense that after a tiger salamander has moved on to land, it doesn't need to have a flat or a tall or flattened tail, so centrum width. It doesn't need to have a flattened tail because it doesn't need to move around in water. Once it's moved on to land, the tail actually gets resorbed. And what you have is sort of what seems to be a much higher ratio of length to width in the centrum. So this, again, just shows us the power of tiger salamanders as a proxy for a lot of information about life history using simple phenotypic traits. Okay, so distributions in body size. And so this is what I first did when I took all of this body size data. And for my purposes, I'm using centrum length here and stratigraphic units on the bottom. And just from first glance, we don't quite see any significant sort of differences in body size. There is one interesting thing. The takeaway point from this graphics is that after, so the main sill, this was about 120,000, about more or less 120,000. Basically, throughout this entire period, the Sangomar interglacial captured by these lithologic intervals, you have the abundance of these really big forms, and these are cannibals. And the fact that cannibals, persisted throughout the interglacial tells us that this pond was permanent and it was deep and it was overcrowded, because overcrowding leads these salamanders to basically become cannibals. So what I did was I took my body size data and I broke it off into different parts to look at some of the central tendencies of body size. And essentially, there's a lot going on here. The takeaway point is I want you to focus on the period between about 120 to about 75. So this period right here, the part where you see all these big changes. And this is really interesting in the sense that during the Sangomar interglacial period, beginning around 120,000 years ago, you have sort of these really, really large metamorphic forms that are much larger, this is mean size, much larger than the neotenics. In terms of maximum size, we also see that these neotenic forms increasingly become much larger than the metamorphics, even though throughout this interval, they've remained consistently smaller than the metamorphics. Metamorphics in green, neotenes in blue. So there's a lot to parse apart in this one graph, simply because all of these different life history traits are being collapsed into one quantifiable trait, which is body size. But my job is to really take this and break it apart and to understand what is going on. Combining all these data, what are these data points telling me about this paleo environment, which is my goal. So I'll move on, but I'll come back to this. So just to summarize all this work and all this data, there's a lot going on. So what's the takeaway point? Takeaway points. So around 130,000 years ago, we have this transition from a metamorphic to a neotenic majority and an increasing neotenic size at the end of the Bow Lake glaciation. We can say from this that there was this transition from a marsh to a lake environment simply because to have such a high neotenic majority, you have to have a lake that doesn't dry out, because otherwise you won't have a habitat that's capable of sustaining all these neotenics. In addition, increasing neotenic size suggests that these neotenes have more time to grow in water, and again, that requires stability of the habitat. We have high terrestrial abundance and attainment of the largest range in metamorphic size, 126 to 115,000 years ago. So that suggests warm terrestrial conditions with a limited but permanent aquatic environment. Now, the fact that the largest range in size is achieved 126 to 115,000 years ago is interesting because, again, under warmer conditions, you have more post-metamorphic growth. And how that manifests in the fossil record is a larger range in size. And this is a really interesting finding in particular because there's been studies that have just come out this year in quaternary research. In particular, there's a study on the fossil insect assemblages at the snowmass site. And they actually found the same thing, that there is this period of really warm conditions about 126 to 115,000 years ago. And that's something that is also supported, if you look at geochemistry, by the work that's been done on the Sangomar interglacial by looking at some of the the geochemistry work that's been done in the basin that suggests that there was a period of peak warmth around this time period in the beginning of the Sangamon. And finally, we have, again, increasing neotenic body size, reestablishment of a neotenic majority, larger metamorphs versus neotenes, and sparse but consistent presence of cannibals throughout the latter half of the Sangamon interglacial. And what all of those tell us is that we have to have had sustained warm and deep aquatic conditions. In addition, there seems to be faster growth after movement onto land. And this is something that could be explained by multiple different things, such as, what if only the largest salamanders move onto land and the small ones, not that the ones that move onto land grow faster, but just that the ones that move onto land are the ones that are already the largest ones. And this last point in particular is really interesting just for the future of this type of project in the sense that there's a lot of neontological studies trying to explain why it is that you have these size differences between different morphological classes. Why is it that you have in some ponds, the neotenes being the largest ones, and in some ponds, the metamorphics being the largest ones. And all of these modern studies are trying to find the mechanisms behind these processes. And what the fossil record allows is the fossil record allows you to use data that preserves a time scale that can never be captured by modern studies to answer these questions. Are the salamanders that moved onto land simply the ones that just happen to be the biggest growing ones? Or were they really growing faster on land? So that's an open question. Okay, and finally, just some comparisons across our records. So like I said, there's not much that's been done in this area. There's a few studies, but what's interesting about our record is that we record a persistent coexistence of neotenes and metamorphs. And that's really interesting because in previous studies, that these forms have all been limited to a designated category in terms of climate or whatnot. Tien 1942 said that neotenes are glacial forms. Rogers 1985 said that neoteny only occurred during the warm interglacial. What our record does is our record shows that these two forms can coexist in the same pond more or less at the same time throughout this 80,000 year interval. And that sort of led us to seek answers. Why is this the case? And what we have been able to say from this is that in Rogers, we know that there were fish in the pond. And we know that to have these neotenic forms, you can't really have fish because fish will just eat up all the neotenes. So what we suggest is that the presence of fish in this pond is the reason why you only have neotenes during the warm interglacial, even though in our study, during the warm interglacial, the metamorphs are abundant and they're also the biggest ones. We find coupling between trends in body size and abundance, which is something that was not found at a Holocene site in Yellowstone that consists of pack rat middens. And in that study, Brusgel et al., there's no correlation between size and abundance. And the reason for that is because multiple populations were sampled. The Yellowstone Lamarck Cave deposit captures a transitional environment that we're sampling from multiple different ponds in the area, whereas our study only sampled one pond. And it makes sense, when you have an environment that allows you to become more abundant, that environment should naturally allow you to grow bigger in size. So there should be a correlation between the two trends. And finally, our population actually records consistently smaller elements than other fossil populations. So when compared to the modern study, Worthington and Wake 1972, the numbers are similar to our study. But when you look at these other fossil populations, Rogers and Brusgel et al., they actually found much larger and consistently larger salamanders, as in the entire the means, the entire frequency count is shifted by 0.2 to 0.5 millimeters in one or the other direction. So we attribute that to sort of just large-scale changes and our characteristics of a population that is highly sensitive to the local environment. And we attribute the small size at our site to the fact that we're in an isolated region with sort of very limited constraints of space. There's only so much space in that one lake to support salamanders, as well as on food source. This is above the snow line. There were microinverts in the pond, but to be honest, we don't really know what they ate or sort of what it was like for them in the pond. So there's a lot of open questions here. And finally, I guess I just want to end with sort of a plug for Ambystoma tigrinum as an ecological proxy. The Ambystoma tigrinum fossil record, more so than allowing us to reconstruct this paleo environment and better understand this high elevation site, it really showed us the power of using vertebrates as an ecological proxy. It allows decoupling of temperature from moisture, which is something that is often that are often confounded in the fossil record. It allows sensitive reconstructions of local climate. And most importantly, for our purposes, it allows paleo-environmental correlations across the entire continent throughout the Quaternary. The Ambystoma tigrinum species came about in the Pliocene and have been prevalent ever since. And by correlating sort of how populations are, by correlating patterns in these populations across the continent, we can really pull together a very cohesive image of what was going on during the late Pleistocene across this climate transition. And all of these answers are going to be important because these salamanders are still around today. And salamanders are in global decline. There's a lot of work in conservation in amphibians, and all of this is relevant work that can really contribute to that effort. So thank you so much.
We have time for one question. Yes, Norm?
Are these types of salamanders toxic?
No, they're mole salamanders. So they're the kind that like dig underground. Yeah.
Okay, thank you, Zixiang.
Thank you.
Okay, so advance if you want a black screen.
Okay.
You go back. You okay, Zixiang?
Okay.
All right, are you sure?
All right.
All right, our next speaker is Annie Osborne, and the title of Annie's talk is Phylogeny and Fishery Management of the Caribbean Spiny Lobster, Palmyra Argus.
Hi. So as the title of my thesis suggests, most of what I did was look at the phylogeny, which is the distinct genetic lineages of the Caribbean spiny lobster, whose scientific name is Palmyra argus, or P. argus, or as I took to calling it in my hours in the lab, private argus. This presentation is going to focus primarily on the phylogeny portion of my thesis, mostly because time is limited and the fishery management just, it's easy to ramble on it. So just to outline briefly what I'm going to be talking about, I'll give you a little bit of background in the sources of my research and my research questions. I will do a series of acknowledgments, etc. And then I will plunge into the life history of this peculiar little critter, P. argus. Then I'll talk about my research methods. I will describe all of the process of the phylogenetic tree and go through all of the exciting statistical models that I ran on my computer, which you will find extremely fun, I assure you. And then I will close with a couple of conservation considerations. And again, those will be kind of towards the end, and if you want me to expand on them, I definitely can. So the basic purpose of this research was to determine whether or not there existed a previously undescribed subpopulation of Palmyra argus in the Caribbean. There are a number of described subpopulations that exist specifically in the Caribbean. There's a more distinct subpopulation that exists even further south in Brazil. Most of them are place-linked. A couple of these subpopulations migrate between several islands, but they're mostly pretty tightly linked to a specific area. And previous research that I and a group of classmates did suggested that there may be a population whose origins we didn't know and whose genetic markers we hadn't identified sufficiently. So that was the main purpose of my research, and as the research continued, I began to explore more potential ways to conserve the Caribbean-wide fishery, because right now it's valued at about $456 million annually, and it's the largest fishery in the Caribbean, including shrimp, which is somewhat surprising. So I began this research with Sea Education Association. It's a program that Stanford has a lot of ties to. I actually didn't do the program that is specifically linked to Stanford. I went to Woods Hole to do my research. And basically, the program takes students on a tall sailing ship from the Caribbean up to Woods Hole, Massachusetts. So general scene setting. The boat is about 170 yards long. It's feet.
70 feet long. It's very, very small and tight. The port side is the science deck, and all of the science equipment to collect specimen like the spiny lobster is deployed over the side of the ship. So almost all of my initial research was done in that little, 20-foot wide space. Super fun. So all of the people that I worked with at SEA are Dr. Amy Sueda, Drs. Eric and Linda Amaral-Zettler. They were my primary mentors over there. I also worked with a number of classmates who helped me develop a lot of these questions, who had a lot of questions of their own that I helped with. So a lot of the fundamental research questions that I was asking came from this particular educational experience. My mentor here is Dr. Paula Waylander, and my second reader for my thesis was Dr. Kathleen Phillips. Thank you guys, both. And I also have to acknowledge, obviously, the School of Earth Sciences and the Earth Systems Department, and a lot of the funding was provided by the Deaton Fund's small grant through the undergraduate advising and research. So, Panelurus argus, the lobster that I'm looking at, is called the Caribbean spiny lobster. It has two main distinct subpopulations, one that is Caribbean-wide, that means from the Florida Keys down to Venezuela, and the other that exists in Brazil. They both have pretty separate fisheries. If you're fishing for the subpopulation that is in Brazil, you're generally not going to be competing with that many people who are fishing up in the Caribbean. And P. argus exhibits source-sink population dynamics, which basically means that some populations in the Caribbean will have more offspring, and some of them will have fewer. The way that lobsters breed is they broadcast a ton of offspring into the current. In their early life stages, the lobsters are planktonic, so they just drift throughout the Caribbean. Depending on whether you're from a source population or a sink population, more of your progeny will either come back to will land in a sink population. So, sorry to say this again. If you are in a source population, and you're a lobster and you're giving birth to progeny, then all of your progeny will most of your progeny will end up in a sink population. If you are in a sink population, then you will be spreading fewer lobsters out into the rest of the Caribbean. So that's basic source-sink population dynamics. And in the Caribbean, there's something called the Pan-Caribbean recruitment theory, which is basically that sources and sinks are linked throughout the Caribbean. So if you are a source in, say, the Dominican Republic, then you could have sinks anywhere up in Florida, you could have sinks in Jamaica or Costa Rica, lots of different places. And also if you are a place like Panama, you could have lobsters coming in from Cuba, wherever. So there are a distinct set of source populations and a distinct set of sink populations. A number of them are associated with their own sub-populations within this Caribbean subpopulation. So my question basically was whether or not there was an unidentified subpopulation in here that could be either accepting a disproportionate number of lobsters or sending out a disproportionate number of lobsters. I wasn't particularly interested whether or not it was a source or a sink, and the place that I ended up getting my individuals from wasn't really determinant of that. I was really just interested in seeing whether the genetic markers had been recorded yet. So what does the Caribbean spiny lobster look like when it is a baby? It is called phylosoma when it's very little. This is what it looks like. It's actually very cool. It looks kind of like it's translucent, pearly white under a microscope. It's really beautiful. You can see all of its internal organs. There are 10 larval stages for the phylosoma, and it takes about 6 to 12 months for it to molt through all of them. It is planktonic for the first 8 stages and remains sort of semi-planktonic and only develops a little bit of swimming ability in its later stages. Starting at around stage 3 or 4, depending on the larvae, it begins to exhibit diurnal vertical migration. And that basically means that since these larvae do not like light, they will swim down the water column during the day and swim up during the night. So on the ship, we would deploy nets twice a day, but we would only get lobsters at night when they swam up the water column. So once they molt through all 10 larval stages, the phyllosoma will become puerily. They'll enter the non-feeding puerulous stage, which means that they have to return to land before they become juveniles. And they can't eat during this time, so the further they are from land, the less likely it is that they'll actually make it, settle on the seafloor, and become juveniles. They basically need to be back on land, or on the seafloor by the time they become juveniles, otherwise they'll starve. So that's basically what the organism looks like. The way that we gathered these organisms was by deploying 3 nets. I said twice a day, but effectively it was once a day. This is a picture, obviously, of the daytime, but this was happening at night under the stars and the moon. The way that we deploy these nets is in a 3-net stack. One net is dragged at 200 meters below the surface. The next net is dragged 100 meters below the surface, and this is dragged so that the square opening is half above and half below the surface. Typically, we would catch lobsters in the middle and bottom nets. Very, very rarely, we would catch them in the top one. And these are all larvae. We would never be catching adult lobsters out at night in the middle of the sea. So we were sailing primarily in the Sargasso Sea, which is a square of the Caribbean Sea that goes from the US Virgin Islands-ish. It moves every year because the ocean is, not land. And it goes up until about Bermuda, a little bit north of it. So the year that we were sailing, the southern boundary was right around St. Croix in the US Virgin Islands. So that's where we collected all of these lobsters, was in this transect of the Sargasso Sea, going again from the Caribbean up towards Woods Hole. Once we had collected the lobsters, we would cut them along the middle. We would store 1/2 for Sea Education Association to keep as a voucher, and then we would dice the other one up very finely to begin doing DNA extraction and amplification. So that was the first iteration of all of the research. When I got back to SEA, I ended up expanding the sample base that we were looking at. I went into their archives because they had all of these half vouchers that other SEA students had left before. I went through all of those, and I ended up choosing about 170 of them. A number of them I couldn't choose because they had either degraded to the point that I couldn't dice them myself anymore, or they'd turned yellow and reacted with something that was accidentally stored with them in ethanol, which happens. So basically, I expanded the data set that SEA had collected, or that our class with SEA had collected, to all of the samples that SEA had previously collected to include in this phylogenetic tree. So once I had chosen which organisms to use, I actually did the extraction and amplification. This was really fun for me because it meant that I got to use a pipetter forever, and I really like that, but it's not so fun for you, so I'll run through it really quickly. Basically, I used the DNeasy gDNA extraction kit. It's a number of elutions that just washes the organism gunk off of the DNA, essentially, is what's happening. Did PCR amplification of the genetic marker that I was actually looking at, which is the hypervariable control region of the mitochondria. It's also called the HVCRD1 gene. I generally just call it the hypervariable region. The reason that we were looking at this gene is that it is hypervariable, which means that it's very easy to tell lobsters apart based on this particular 760 roughly base pair area of the DNA. So the gene sequencing ended up being done at the Keck Ecological and Evolutionary Genetics Facility in Woods Hole. I got all of the sequences sent out to me by about February, and then I spent like 3 months just editing them down because there were lots of gaps. The older the organisms are, the more degraded they tend to be. So that was a lot of time that I won't discuss. The next thing that I ended up doing was doing a BLAST database search. This is an intranet database of pretty much all the previously stored DNA sequences for all organisms including the Caribbean spiny lobster. So I would search every single sequence to see if there was a matching previously recorded organism, and if there was, then I would include that in my phylogenetic tree to see how closely it related to other organisms. So this was just a way to make sure that I wasn't just like randomly putting together a tree of a bunch of organisms whose relations to existing ones I didn't understand. So that was the purpose of the database search. Then I used the Mega software alignment tool to do my phylogenetic tree. And then I used Mega and Atoll to create a tree that people could actually look at. So the tree that I have, this is the prettier one. It's circular. There are about 170 organisms on this tree, which means that there's pretty much no way I can show it to you where you'll be able to read all the names. So here's the condensed tree with all the information that actually matters. What matters is that only 5 organisms were separate from the rest of the organisms by any relevant metric. So there were over 160 organisms that were so close to each other that the bootstrap values on the tree were like 0 to 5. So they were incredibly closely related. There seems to be virtually no genetic distinctions between them and previously distinguished populations. These ones were different by Kimura 2 parameter values of greater than 0.05, which is the generally accepted value to separate genetic lineage groups in the Panolaris argus genetic world. Basically, the Kimura 2 parameter is a statistic that measures switching purines and pyrimidines in the genetic, in the DNA. So there were 5 of them. That was about 3% of my data set. A previous data set that we had looked at, there were about 5% that didn't fit in. So now expanding this data set to include about 170, the percentages dropped. And it seems like there is really no distinct subpopulation in the Caribbean. So that was mostly what my researchers found, which means that I don't have a ton of applications to the real world from this particular portion of my research, but I still have a lot of thoughts on fishery conservation management. So I'm going to talk about it just a little bit now. Basically, coral reefs are the habitat for adult lobster, whom I haven't talked about that much because I was looking at the little larvae. Coral reefs, as a number of presentations already today have talked about, are in serious trouble from a number of threats. One of the biggest ones is ocean acidification, which I'm not going to touch on because lots of other people, I think, have talked about that. But there are a lot of upstream problems with coral reefs in the Caribbean and throughout the world, but specifically in the Caribbean, which include sedimentation and eutrophication from logging, coastal development, and agriculture. There are also some local issues with coral reefs that are specifically attributable to lobster fisheries, specifically fishing gear, which can break loose, can either be cut loose by fishing competitors, can break on its own, or can get cut off by ships that are going through shipping lanes that fishermen are also using. So basically, the primary suggestion that I have from looking at this is that fishermen need to have more fishermen need to get together into cooperatives so that they have more representation at different levels of governance. For point source management, there needs to be cooperation among different territorial fishing groups because a lot of the problem that's happening with things like fishing gear is people from different fishing communities will go out and cut people's traps, and that sends the trap sort of basically careening through the coral reefs, and it does a lot of physical damage. There's also a specific type of fishing gear called the casita, which is basically just trash that people throw onto the ocean floor, and lobsters congregate under it because it resembles a coral reef. But the problem is that they can also get moved around a lot by ocean currents, and because they're things like sheet metal and lead and pipes and old tires, they can do a lot of physical damage to the coral reefs. So the more organization among fishermen that there is, the less likely that this type of damage is to happen to coral reefs from these sort of point source issues. In terms of upstream management, Because the damage that's being done is kind of nebulous and can be attributed to a number of different companies, a number of different economic drivers, it's really hard to say in the amount of time that I have all the things that can be done to mitigate some of these issues. But again, if fishermen have stronger intercooperation, then one of the things that they'll be able to do is get more leverage in representing themselves to a lot of the economic interests that represent logging, coastal development, agriculture, these types of things. And they can also coordinate with people like the tourism industry, who depend a lot on coral reef safety, to do a lot of negotiating with people who are upstream polluters. So cooperative fishing is going to be a very important part in stepping forward to manage the fishery as it continues to develop in the Caribbean. The good news is that the lobsters are not particularly overfished, which is not something that you can say about a lot of the other fisheries in the Caribbean. So it's really more habitat protection that needs to be done. Sorry, this did start at 4:10 in the end, right?
Yeah,
Am I? Okay. Sorry. Okay, that's mostly what I have to say. Sorry, I rushed that one a little bit, but do you guys have any questions possibly about that? Anything earlier than I ended up saying?
Yeah, we have time for a couple of questions for Annie. Ms. Katie?
So you talked about these lobster populations being either a source or a sink in the Caribbean.
Mm-hmm.
What does that imply in terms of what we need to think about for their conservation?
Yeah.
So I guess repeat the question. What is the idea of source of sink populations imply about lobster conservation?
So it implies a sense of responsibility to nations that have sink populations, and it also implies that there needs to be some sort of protection for nations with source populations. So if you think about it, if you are a nation or Cuba's got both, but like a number of the Bahama Islands are source populations. So fishing communities there would need to have some sort of leverage to say that their populations are particularly important. They're not just important for their own economic, their value to the local fishing populations. They're also important to fishing populations throughout the Caribbean. So that might mean some international cooperation if there were upstream pollution from different countries that was affecting these source populations. It might just mean higher level governance working on these issues. But also sink populations, there are a couple of implications for sink populations. On the one hand, it actually gives the fishing community and upstream polluters a little bit of license to pollute because those populations aren't so important. But on the other hand, it means that more people could potentially benefit from those resources because more people can harvest them without serious ecological damage to the rest of the populations throughout the Caribbean. So.
Thank you very much, Annie.
Yeah.
We should probably move on to our next speaker. Okay, our next speaker is Linta Reji. Linta's project title is Characterizing Keeler Cageings in Estuary Sediments of San Francisco Bay.
Okay. Hello, everyone. I'm not going to repeat anything that Richard just said, so let's just get right into the talk. Here is a roadmap of my talk today. I'm going to give you some background on my topic and then present you my research question, after which I will explain what I did, how I did it, and what I found out. And then we will end with the major takeaway points as well as some future directions. So first, some background. I want to start with estuaries. As we all know, estuaries are systems that form the transition zone between terrestrial and marine environments. That's where freshwater and saltwater mix. And these are also regions with really high biodiversity. And when estuaries are particularly located beside population centers, they get a lot of nutrient input, and this increased nutrient pollution can actually lead to these undesirable consequences, including eutrophication, hypoxia, and harmful algal blooms, none of which are good for estuarine health. So that's why we worry about nutrient pollution and having excess nutrients in these estuarine systems. One way in which we can get rid of these extra nutrients in the system is through microbial nutrient cycling. For example, the excess nitrogen that is in the system can be metabolized by microbial organisms and can get converted into nitrogen gas, which can then just escape from the system. This is what actually happens inside this box, which is the marine microbial nitrogen cycling. Now, I know that this figure is probably not the most inviting, but I just want you to look at just two of the major features here, which are nitrification and denitrification. So nitrification is the process that converts organic nitrogen into a species, a chemical form, nitrate, which can be taken up by denitrifiers and convert into nitrogen gas, which then leaves the system. So if we take a closer look at what's actually going on, we have ammonia that enters the nitrification stage, gets converted into nitrate, which then gets denitrified into nitrogen gas, which escapes the system. So we also have two different communities of microbial organisms that mediate each of these processes. We have nitrifiers here and denitrifiers here. Now, this is not as simple as it is presented here. So if we take a closer look still of nitrification, it's actually a two-step process. We have ammonia oxidation and nitrite oxidation. So what actually happens is ammonia enters the ammonia oxidation stage, and we get nitrite. Nitrite then gets oxidized into nitrate. And we again have two different microbial communities mediating each of these steps. We have ammonia oxidizers here and nitrite oxidizers here. So this is the picture of nitrification that we had for over a century. This is what we thought was happening in the environment. We had ammonia oxidizing bacteria that converted ammonia into nitrite, and then we had nitrite oxidizing bacteria. For about 10 years ago, I guess, there was this finding that suggested that another group of microorganisms could actually be able to do this top process here, ammonia oxidation, and those organisms are archaea. Now, just to remind us what archaea are, these are single-celled microorganisms that are found in many environments, and they're actually, in evolutionary terms, they are really distant from both bacteria and eukarya, which include us, of course, that they form the third domain of life here. So now we know for sure that archaea are indeed involved in ammonia oxidation, and we call them ammonia oxidizing archaea. I'm going to refer to them as AOA from now onwards, just for convenience. So it's often observed that these organisms are really abundant, and in some environments they actually are more abundant than their bacterial counterparts. But it's a pain to grow them in the lab because they are such slow growers, and because of that we have to explore potential molecular methods to look at them and study about them in the environment. That's why we are interested in finding potential genes that could work as biomarkers for these organisms in the environment. And the conventionally used gene for studying about these organisms is the AMOA gene, ammonia monooxygenase, which is the gene that codes for the enzyme that catalyzes ammonia oxidation. Now, recently, we've also observed that another gene in their genome, nitrite reductase or NRK, is also highly abundant and highly expressed in many environmental systems. But the problem is we don't know what this gene is doing in these organisms, and we don't really understand the ecology and also the diversity of this gene. And there have been very few studies looking at the ecology of this gene so far. That leads me to my research objective, which was to investigate the abundance and diversity of archaeal NRK genes across San Francisco Bay Estuary, specifically in sediment samples. Here is a map of San Francisco Bay. I had 26 different sampling stations, and I did not collect any of these samples. I used archived samples that Julian in the Francis lab collected. Yeah, as I was saying, I had 26 different sampling stations, and this part of the bay I'll refer to as South Bay. Here we have Central Bay, then North Bay, then 2 riverine stations up here. Okay, now what I did and how I did it, what I found out. So I did 2 different things. Basically, I looked at the abundance and the diversity of the gene. First, the abundance. Basically quantified our Kellner-K copies in these 26 samples. And we have an assay called qPCR that we use for quantifying gene copies. And if any of you want to know more about this method, please ask me after the talk. So this is what I found out. Here on the x-axis, we have all the sampling stations going from South Bay to the North Bay, and eventually to the riverine stations. And on the y-axis, we have copy numbers per gram of sediment, and it's in logarithmic scale. You can see there is quite a variability in copy numbers across the bay, particularly in the 2 riverine stations. The abundance is much lower compared to the other stations. But even more interesting is when I plot AMOA numbers with the Kellner-K numbers. You can see there is such a huge incongruity between these 2 gene copy numbers. There are several potential explanations for this, but this is what I think is happening based on my sequence data. The traditional AMO-based assay could be underestimating the actual AOA diversity. Now, if you want to learn more about the other possibilities or discuss them with me, I'm more than happy to do so after the presentation. So now, moving on to diversity. It basically involves cloning and sequencing, and this is sort of what we get out after sequencing. And you may recognize this as a DNA-based sequence. So looking at diversity basically means comparing a bunch of these sequences and establishing how different or similar they are based on base composition. And in microbial ecology, often we want to also define something called operational taxonomic unit, which is analogous to species. We do this because it's really hard to predict when 2 sequences are coming from the same organisms or from same species or from different species. That's why we often define something called OTU. And in this project, I put my similarity cutoff at 97%, which means if 2 sequences are 97% similar to each other, then I treat them as 1 OTU. Next thing I did was something that Annie just explained, put these OTUs in a phylogenetic tree, and I also used MEGA. So I know that this tree is not readable, and I don't want you to even try reading it, but just look at the colors. We have blue here representing sequences from the South Bay, red represents Central Bay, green North Bay, and there is some purple somewhere here, that's riverine sequences. And the black sequences come from the literature, those are the reference sequences. So looking at this tree, you can see how the colors are kind of all over the tree rather than being concentrated in the specific clusters, which suggests that the community, the diversity is not that variant across the sampling stations that I was looking at. Now, if you look at this highlighted part down here, you can see how a little too much red is here. And what's interesting is that all of these sequences come from the exact same site, all these red sequences. That's a Central Bay site, CB129S. And especially in this branch, there are 29 sequences that fall into that OTU group. Now, I wanted to look at how variant all these stations are based on community composition, and for that I used something called ordination method. So this is a way of looking at similarity or differences between samples. So points that plot close together in a PCA plot, this is a PCA plot by the way, those points are more similar to each other than points that plot far away. So here, the site that I was just talking about, CB129S, plots really far away from the other stations, which suggests that the community composition there is actually quite variant from the other stations. Now, this plot doesn't tell me anything really about what is causing this difference, and I wanted to know if some environmental parameters could be influencing this community structure. For that, I used another ordination method called canonical correspondence analysis, or CCA, which is, you read this plot just the way you read the other plot that we just looked at, but the only difference here is that we have these arrows, which are the significant environmental variables that are significantly affecting community composition here. And I have total organic carbon and dissolved inorganic nitrogen as the significant environmental variables. Again, you can see how CB129S is really far away from all the other stations. And again, there is the South Bay side BA10, which also floats quite far. And another side is BG20. Now, what could be going on here? I plotted the TAN and TOC concentrations in the sampling stations. Here is the dissolved inorganic nitrogen graph. You can see how it's really, really high in BA10 compared to all the other sampling stations. So that could be causing what we just saw in the plot earlier. Now, if you look at CB129S, it has one of the lowest concentrations of TAN. Now, total organic carbon, both CB129S and BG20, the third side that plotted away from the other stations, they have the lowest TOC concentrations. So this is suggesting that maybe TOC is affecting the community composition in these stations. Now, I wanted to also look at, if I take out these outliers, do I get better resolution and better idea of the community composition in the other stations? So that's what I did next. I took out CB129S and BA10 from the analysis and did a CCA again. And that suggested that only total organic carbon is the significant environmental variable that's affecting community composition here. Sorry. So that's this arrow here. Yeah, so TOC could actually represent a source of ammonia for these organisms, or it can also be a substrate for the mixotrophic growth of AOA in these environments. Now, everything that I just said need to be understood on the basis of this plot. This is how much diversity have I covered through this project. Okay, so this is a rarefaction curve which tells us something about the sampling intensity. If I keep on sampling, for example, in this station and go, I had 455 sequences. If I go above that, do I get better coverage? Do I get more diversity that is there in the environment? That's what this plot is telling us. We would ideally want to see these curves plateauing at some point, which suggests that these, the entire diversity is covered and more sampling would not particularly yield any new sequences. But this curve doesn't really plateau, which suggests that a lot of diversity could still be there that is not being covered in this study. Yeah, so that's about diversity coverage, and that brings me to my conclusions. As I was telling earlier, higher abundance of archaeal NRK. This could suggest that conventional AMOA-based assay could be underestimating AOA diversity, which could mean that maybe NRK could be a molecular marker gene for AOA in the environment. The community composition was mostly similar across the bay, but as we saw in those plots, there was some variations as well. And DAN and TOC were important in community composition, particularly TOC, as we saw in the later CCA plot. Some of the future directions, we obviously need better diversity coverage, and also we need to establish linkages between AMOA and archaeal NRK. And we need to get identify more ecotypes of this gene from various environments, including soil, because in this study I wasn't looking at soil sequences. Now, before I end, this is probably the most important part of my talk today, which is to thank people who have helped me do this project. First, I want to thank my advisor, Professor Chris Francis, who was extremely helpful in for his continued guidance and support throughout this project, and his financial support, as well as his really valuable feedback on my draft, which really helped me a lot in revising the draft. And my second reader was Professor Karen Casiodi, who is not here today, but her comments were also immensely helpful in revising my draft, and also she was really, really awesome. And the person I really want to thank again is Jessica. She is the best mentor anyone can hope to have. She's absolutely great. I've learned so much from her, and I feel lucky that I could get to know her. So another person in the lab who was Julian, who is not here today, but he was kind enough to share his sequences samples with me and also some of his data, which was very important in analyzing my results. And thank you, Richard, and the CSER program, because this project was actually a CSER program that I did, the CSER project that I did last summer. So thank you, everyone, for your time. Thank you. If you have any questions, please.
Thank you, Linta. We have time for 2 questions.
Yeah.
So you say you need to get better diversity coverage. Would you do that by getting more samples or by redesigning your PCR you need to pick up?
So I'm just going to repeat the question. It relates to how you could get or improve the diversity of your sampling.
Yeah, so the plot that I had here was for the entire clone library put together. But if you look at the individual clone libraries, those are even worse. I mean, like the diversity coverage is not quite complete. So you were asking?
How would you get better diversity? Would you sample more, take more samples? Somehow redesign the PCR so you can pick up things?
I guess redesigning the PCR would be a better option there rather than, because we already have like 26 sampling stations that spans the entire bay. But even with the PCR method that we do have, we are not picking up all the diversity there. Maybe, but so I was talking to Professor earlier and he suggested that, so we used 2 different primer sets for picking up the sequences, and pairing the forward primer of the first primer set with the reverse primer of the second primer set could pick up more sequences. So yeah, redesigning PCR would be the better option.
Any more questions for Linta? All right, thank you so much, Linta. So please, our session today, I talked a little bit about the importance of friends and supporting us in the senior honors thesis, and I see it's been very important for Aldric here. So Aldric Ulep is going to be talking about his work on water conservation projects in Washington. The title of his talk is "What Are They Doing Right in Yakima? A qualitative study in collaborative watershed planning of the Yakima River Basin Integrated Water Resource Management Plan."
Great, thank you, Richard. Hi, everybody. So, good afternoon. My name is Aldric, and I'm excited to share with you what I've been working on for the past year with the Woods Institute for the Environment. So, the title of my talk, again, deals with this Yakima River Basin Integrated Water Resources Management Plan, or the Integrated Plan. And the group that created this plan was actually nationally recognized and won an award for being very collaborative. And it's been a model of water, or it's been touted as a model of watershed management across the West. But it's also been made up of a bunch of old enemies who've been bickering at each other for decades. And I'm wondering how they came to this consensus agreement. Or in other words, what led to the agreement to adopt the Yakima Integrated Plan? So to do this, I will first look at the context, or what happened before the plan's adoption. Then I will briefly describe my methods, or how my study attempts to explain what led to the agreement to adopt the plan. Then finally, I will discuss my findings, or what insights my study provides. So first off, does anyone know where Yakima is? Where the Yakima River Basin?
Washington State on the Columbia Salt.
Yeah, that's exactly right, in the middle of Washington. Are you from Washington, or?
No, it's just there's kind of folds there that for planetary science look like kind of the structures on the moon, so pictures of it get shown a lot in planetary science meetings.
It's kind of cool. Right, so this is the Yakima River Basin, and it supports a multi-billion-dollar agriculture-based economy. So it's very important to the state of Washington. Can anyone guess what crop they produce? It's so important.
Potatoes?
That's actually apples. So 60%, or almost 2/3 of the nation's apples are produced in this little basin. And so obviously, water supply in the Yakima River Basin is super important because it produces all our apples, and gives so much money to the state of Washington, but it's threatened by two things in particular. The first is over-appropriation. This is when there are more water rights than there is actually water present in the basin, and that's due to the messy nature of Western water rights. The second is excessive drought that's been happening all across the 20th century. And so during a water shortage, some farmers may get as little as 70 to 30% of the water that they have a right to, and it significantly impacts the crop yields and the profits that they make. It's not just agriculture that's affected by the water shortages in the basin. There are a bunch of different other uses of waters. For example, there are out-of-stream uses which require the water to be withdrawn from the river. So you have irrigation here. And there are also a bunch of in-stream requirements. So for example, fish and wildlife need water in the river to exist. And so all of these different water users compete over the same amount of water supply. And so when there's a water shortage, the results can be disastrous. 2 species of salmon have gone extinct in the basin. Now, different stakeholders have different levels of interest in all of these different water uses, right? The group that created this plan is made up of all of these different stakeholders, so representatives from who care about fish versus those who care about their farms. Who is this group? This group is the Yakima River Basin Water Enhancement Project Work Group, or the YARBWEP Work Group. I know it has a funny name. It comprises representatives from all these different stakeholder groups. So you have the federal agency, which is the Bureau of Reclamation, the state agency, which is the Department of Ecology, and these two lead the work group. You also have the local government, so the 4 counties and the city of Yakima. You have the tribal government, so the Yakima Indian Nation, and various irrigation interests, like the farmers, and environmental interests who are concerned with species of fish. And like I said, each of these actors have a different set of interests. Together, the plan addresses all of these different interests. These are the different plan elements. So fish passage, fish habitat enhancement, structure modification, surface and groundwater storage, water markets, which is trading water rights, and enhanced water conservation. The plan is a consensus agreement that all of these elements are necessary for everybody to meet the goals of the work group. And like I said, there were previous attempts to solve the water shortage in the Yakima Basin. This is a timeline of a bunch of the different things that have happened. This orange up here are drought years. And the timeline goes from 1977 to the present. So in 1979, right after a bunch of drought years, the YARBWEP feasibility study authorization was started. And basically it gave federal money. It allocated that to study fish passage in the Yakima Basin because all of the dams in the basin don't have fish passage, which is why all the salmon are going extinct, and water conservation opportunities. Throughout the 90s and the 2000s, so down here, local interests created different plans. So business interests formed this Yakima River Watershed Council. There was a county management plan, etc. But they all fell apart due to one reason or another. Maybe they lost funding or the stakeholder support was not broad enough. In 2003, up here, there were powerful lobbyists that got congressional authorization to conduct a study that only studied storage-based options. So it's called the storage study. And in particular, there was a focus on this one project, which was a multi-billion-dollar project called the Black Rock Dam project. And this group really thought it was the solution. But it was found in 2006 that Black Rock was too expensive and risky and contamination spread. And so the State Department of Ecology started this integrated alternative study And after some local input, the final integrated alternative EIS, Environmental Impact Statement, was released. And from there, Reclamation and Ecology, the federal and the state leads on the projects, invited stakeholders on the work group. And that's this red line, 2009, when the work group was created. And by 2011, consensus agreement for the integrated plan was reached. So only after 2 years, compared to the decades of litigation and fighting that everyone's been going on in this basin. So in light of all of this, another way to rephrase my research question would be, past efforts were insufficient. What was different about the integrated plan? What was different about the Yarb-Webb workgroup process? What changed this time around? And so to answer this question, I looked up literature on various successful watershed planning groups. In particular, I focus on this one meta-analysis study looking at the successful watershed planning groups, Leach and Pelkey 2001. They list factors such as funding, effective facilitation, and broad inclusive membership as important factors for a watershed planning group to succeed. These help me design my methods. I use a qualitative approach to understand the planning process from each of these stakeholders' perspectives and how the planning process really arose. Using factors from the literature, I designed a semi-structured interview. And after reaching out to various stakeholders, I conducted, transcribed, and coded these interviews. These are the interviews I got. These are the people I got to interview, about 14 of them, all from different stakeholder groups and from different organizations. And this is what NVivo looks like, which is the software I use to code each of my transcripts. On the left, you can see this is a sample of my transcripts. And on the right, these are codes. These codes come from the literature, so Leach and Pelkey, and also from the data itself. Through iterative analysis, I organized this data to draw conclusions. After all of that, I identify 3 findings. So my 3 findings are, 1, grassroots groups united on their shared values to move forward. And after that, state and federal leaders pushed their initiative forward. And finally, the work group itself was committed to this balanced approach. And now I will delve into each of these findings by presenting key quotes that demonstrate each of these. So 2 stakeholders, the Rosa Irrigation District and the Yakima Indian Nation, or Rosa and Yakima, have fought over water use in court and do not have a friendly relationship by any means. They talked about frustration over the lack of process, and in desperation, they finally reached out to one another during the storage study, which they both did not like. We came together and we both agreed we can't go forward with that proposal called Black Rock. This letter, they listed mutual concerns. Our joint letter to Reclamation and Ecology was co-signed, and that shocked the Valley. That was a game changer, I guess, is how they call it around here. This letter bridged the irrigators and the fish interests in the basin. From the Rosa's point of view, we realized that our interests were more similar than different. Together, we could convey an awful lot of power. And so we could see that they created power in allyship with one another. The sense of urgency over the drought and climate change and all these water shortages, all the fish dying, and the frustration over this lack of progress led to one of them to reach out to each other and find out their shared preference for a better alternative to the storage study. And they both recognized that there was power in collaboration. They didn't just ally with each other, though. They allied with the state and federal governments. The government finally listened to their local input, like the joint letter. We got leadership at Reclamation and leadership at Ecology to agree with us, and that's when things started to finally rapidly change. They too eventually realized that the storage study was too narrow and that there had to be a better, more comprehensive solution. We had the right political leadership. So this would be the state governor, the state Congress members, the state agencies, and the Congress members in US Congress. They all supported moving forward on what we called the integrated approach to solving these problems. Another state initiative that helped move this along was the state water rights adjudication. So what an adjudication is it basically clarifies the rights that each of the rights holders has to the water in the basin. And what that did was it reduced the conflict because what was once over-appropriated, the conflict is now mitigated a little bit. And at that point, everyone was more comfortable in talking about the solutions, knowing what their entitlements were, or their poker chips, so to speak. So state and federal leaders allied themselves with grassroots groups, and they also readily started implementation. So Governor Jay Inslee, he, in his first act as governor, he actually passed this bill, Water Jobs and Fish Bill, which was implementation for the plan, and it gave $132 million towards implementation. Finally, so you had the grassroots leaders, you had the federal and state leaders. They were all in this work group together, and there was this constant emphasis on a balanced approach from all of the stakeholders I interviewed. They're getting enough out of the plan without sacrificing too much of what they value to push it forward. Here's another quote from the county governor, the county representative. I think the real key to it is that everything moves together proportionally and incrementally. We're not putting one interest in front of the other, and that there seems to be a real commitment to that. Not only that, all workgroup members respected each other's goals and recognized their legitimacies and supported them even if they would traditionally be against it. For example, you have an environmental representative on the group, but one of the elements is dams and increased expansion of storage, which is something an environmentalist would not usually be for. So everyone was willing to compromise, and despite that, it was helpful that Reclamation and Ecology consciously designed the work group in order to select stakeholders who had a dog in the fight, so to speak, but also recognized the integrated nature of the plan. And so some local environmental stakeholders were not included in the work group because the Reclamation and Ecology saw that their only aim was to stop the project at all costs. And so overall, the workgroup planning process served as a discussion forum to express shared values and mutual concerns. And it also, what it did was it joined previous existing efforts to give a sense of ownership to everybody. So you can see up here, these were all federal and state initiatives, and these were all local Yakima Nation or irrigators initiatives. But when the work group came together, that was when all of these actions started coming together and traction started gaining towards this integrated plan. There are several takeaways for watershed planning groups. And that is to involve a variety of stakeholders, to create relationships along shared values, and to emphasize this balanced approach. They might sound obvious, but it helps to prioritize what watershed planning groups should do when they develop a management plan and when they go through this process. Finally, some of what happened in Yakima was actually luck. I feel that there were allies in state and federal government, but you can't really change that easily. But overall, I personally believe that this case study has implications for negotiations and collaborative policymaking in other settings. Thank you, and thank you to all my friends who came. This is really overwhelming. Thank you so much.
Thank you, Aldric. We have time for a couple of questions.
So you mentioned that the two of the groups both did not like the Black Rock initiative.
Mm-hmm.
But so what about it was so bad, and did they change that material for this new plan?
So if I could repeat the question, so what was the opposition toward the Black Rock initiative, and what was changed in the plan to deal with that dislike?
Mm-hmm. A lot of the critics to the Black Rock plan said that it was much too expensive. It was a multi-billion-dollar project, and it was very risky in that the water, it would involve an inter-basin transfer from the Columbia River to the Yakima River, and that would run along the Hanford Nuclear Reservation, and so there was risk of contamination spreading. But the Basin Storage Alliance was still staunch that this is the right project. They still are to this day.
So usually when you have such divergent, powerful interest groups that are against each other, there's going to be one or two key players, key negotiators with experienced backgrounds and bringing these groups together at the table and sort of driving consensus. Were there a couple of key players, to your knowledge, that were the impetus to kind of bring these warring sides together to establish consensus, or was it something more organic than that?
So, yes, there was definitely the reclamation and the ecology leaders who were very experienced. They were both seasoned veterans, so to speak, and so they knew a lot of the history in the basin. They knew a lot of the previous efforts, and they were the ones who really carefully selected the stakeholder groups in order to form the Yarb Wet Work Group.
Okay, thank you, Aldric. We're going to move on to our last speaker. I hope you'll stay for the last talk and for our last speaker, because we're going to celebrate and toast all of our senior honors theses writers today with a glass of champagne subsequently.
Thanks, Kevin.
Take a deep breath. Okay, our last speaker is Nicole Rodriguez, and the title of her talk is Native Hawaiian Healing in the Context of Western Medicine.
E ho mai ka ike mai luna mai e ona mea huna noea ona mele e ho mai e ho mai e ho mai e ho mai ka ike mai luna mai e ona mea huna noea ona mele e ho mai e ho mai e ho mai e ho mai ka ike mai luna mai e ona mea huna noea ona mele e ho mai e ho mai e ho mai e. E ho mai is a traditional Hawaiian chant used to introduce people to nature. It's meant to help connect people to the land. It's used before an exchange of knowledge. It's used as an introduction, and because my talk largely focuses on the importance of protocol and traditional Hawaiian values and respecting those values, I thought it would be fitting to begin with that chant. So, my research is on investigating Native Hawaiian healing in the context of Western medicine and public health. So, a core belief of Hawaiian traditional healing and Native Hawaiians is that the health of the land is the health of the people. Environmental degradation and the degradation of resources in the area right now have led to a transformation of the healing culture of the islands. Western society has not only influenced the ecosystems in Hawaii, it's affected the way that indigenous people communicate and the way that they're functioning right now in Hawaii. It's affected their health. In Hawaiian culture, health and well-being is rooted in a strong connection to environment and balance. A fundamental theme in Hawaiian health is the idea of being pono. Pono does not have a direct English translation. Pono, it means being righteous, being good, being respectful and balanced. It means living in a way in which your values are aligned with your actions, your thoughts, and your feelings. To be pono is to be one with everything, to live right, live well, and connect in a way that your values, morals, and ideals are all in harmony. And this is at the root of Hawaiian traditional healing. Central to the foundation of Hawaiian healing is the concept of lokahi. Lokahi means balance, harmony. For a community to have health, there has to be a balance between the human component, the spiritual world, and the land. Kanaka is man, Akua, God, the higher power, and Aina is land. This is also connected on the individual level. For someone to lead a healthy life, they have to have balance in mental health, spiritual health, and physical health. A person cannot have physical health, according to Hawaiian beliefs, without having mental health and spiritual health. The purpose of this study is to investigate the cultural gap between indigenous healthcare and Western healthcare, and to explore the ways to effectively integrate both of these modalities. The problem right now is that the healthcare system is facing right now is that these are completely different medical models. To the left is my friend Dane Silva. He is one of the most renowned practitioners in Hawaii in the traditional healing community. And for a patient seeking healthcare, they have a completely different experience visiting a healer like Dane than going to a Western healthcare model. And these two different viewpoints on health and on life lead to a major cultural disparity. An overarching theme right now in the Native Hawaiian academic community is that we have to dive into our past in order to learn how to build our future. In this particular case, in order to learn how to address the needs of the community in Hawaii, it's important to institute preventative health care that tailors for Native Hawaiians. It's a Hawaiian society that's been overtaken by Western culture. The way Hawaiians see it, they've been uprooted from their land, uprooted from their culture, and from their fundamental right to health. So data collected while researching for this thesis led me to believe that as a result of Western influence and social change, the healthcare system in Hawaii has evolved in a way that does not meet the needs of the Native Hawaiian community. Key reasons include my two main hypotheses. One of the reasons that the Western healthcare does not meet the needs of the Native Hawaiian community is that it does not reflect the key values of this traditional healing. This goes back to our model with the balance of mind, body, and spirit, but it also relates to them targeting initial well-being instead of the prescriptive medicine that we have today. Western doctors are also not necessarily tuned into these cultural preferences, and clinics do not provide traditional healing options or refer patients to traditional healers that they'd like to talk to. And my second hypothesis has to do with the the first hypothesis was about the fundamental theory behind medicine, and the second one is kind of the institutional practice of medicine, so theoretical and in practice. And for this one of the reasons that Western healthcare does not meet the needs of the Native Hawaiian community is built into the fundamental structure of the way it's set up right now. There's limited exposure to traditional Hawaiian culture within the system, and there's also little recognition of the practices. So Native Hawaiians are essentially forced to go outside of their culture and into a foreign culture, the Western culture, to seek health care. And this goes against the traditional Native beliefs of going within their own family, within their own society, and within their own culture to seek help. So data were collected from 22 open-ended interviews in Kauai, Oahu, and Big Island. Samples included healers, public health officials, practitioners, physicians, ethnobotanists, and researchers. The interviews were selected based on initial Stanford references through faculty and students, and hospital communications, and also snowball sampling. Every time I met someone, they had like 5 other people they wanted me to talk to. And I coded everything with in vivo, as Aldric beautifully illustrated earlier with his presentation. And my results show that Western health does not meet the need of the Native Hawaiian community. And let me see, sorry. And this is because it does not present the values. And the 3 main values that I distilled were the value of spirituality. For them, healing is 80% spiritual and 20% physical. So it is healing. Like, you heal through your spiritual health and through your connection with God or whatever it is. It doesn't matter. The point is that you have that component in your life. And the value of community. Your relationships in terms of like Hawaiian healing are absolutely key. Your family, your community, the people around you, and if that is healthy, that allows you to be healthy. And lastly, the value of environment. Their connection to the ocean and to the land is crucial in having a healthy lifestyle. And these are a few of my favorite quotes that I wanted to share with you guys to back up some of my statements. I guess I'll read them aloud. Western medicine is focused on addressing disease, while Hawaiian healing is focused on establishing wellness. It looks at keeping the body strong instead of addressing a particular illness. La'au is not like Western medicine. Akua does the healing. La'au is traditional herbal healing, and Akua is the higher power. Making changes spiritually starts manifesting physically. So everything goes back to the spiritual health. Healing is not about the medicine coming to you. It's about you going to the medicine. You're engaged in your own healing. Hawaiians heal differently. It's a very different mindset. It's very collaborative. It's the native mindset. Together we figure out what can be done. And some of the in vivo things I wanted to mention. So 100% of the healers suggested that spirituality was absolutely key. They all backed up that the value of family and community was crucial for health, and that nature was central to healing. So everybody agreed. The quotes that I mentioned earlier are just very eloquent, representative quotes that represent everything that people have been talking about. And though there was a range of cultural understanding among physicians, all of them showed interest in learning about Native Hawaiian healing and how to deal with these problems. So the desire from the medical community is there. The doctors do want to learn how to deal with these issues. But one of the main problems is the structural issues that we're facing in the public health system. Which leads me to one of my second set of results, that in addition to not reflecting Hawaiian values at the core theory, there's a minimal influence of traditional Hawaiian culture in practice in Western Health Center. So what we have right now is that physicians are rarely focusing on the patient holistically in the context of family and community, and that the time commitment that they're dedicating to patients simply is not enough to develop the strong relationship and the trust that Native Hawaiians are accustomed to in sharing all of their health needs and concerns. Like a regular 20-minute visit will not do for a Native Hawaiian. They're just not going to open up, especially when they come with this background kind of mistrust of Western culture because of everything that they've done historically to the society. So there's a lot of things that have to be amended for when working with these Native populations. And lastly, they have a really limited baseline knowledge and understanding of Native Hawaiian practices. So whether or not they want to, they can't really relate to these patients because they don't have that background. And one of the reasons that Native Hawaiians are disproportionately unhealthy, it's because they're actually avoiding health. They don't seek out the health care. The traditional indigenous health model in Hawaii actually addresses these social determinants. So the way that healing was done historically, and is still being done in small communities, they target all the determinants that are affecting their health right now. They target problems in the family, problems spiritually, issues with education, and they really address everything to give people that strong foundational health. So more interviews that support these statements. This is from the North Hawaii Community Health Center, which is one of the few centers that's working to integrate Western health and indigenous healthcare. And their goal is to hit the Native Hawaiian population because they don't want to come to the doctors. They don't trust the mainland doctors. Most of medicine is communication. Not understanding culture is poor communication. And something that came up repeatedly is that mental health and behavioral health are important to always consider in an integrated approach. So you don't just address a physical ailment, you talk to their patient about their mental issues, emotional issues, spiritual issues when you're talking to them about this specific concern that they're coming to you for. And getting help within your culture is a helpful thing to do, and it's important to face for the face of the clinic to reflect the face of the community. So this is especially important for Hawaiian communities and just minorities in general. It's nice to have your community and your clinic be one and the same so that you feel comfortable. So the results overall suggest that clinics and hospitals need to be restructured in order to emphasize cultural competency and provide patients with culturally appropriate treatment throughout the islands. And more than anything, give patients the option of whether or not they want to choose Western healthcare or indigenous healing practices, because right now they don't have that option. And my results also suggest that the healer-patient relationships need to be restructured in order to increase access for the patients to have this healthcare option and to empower the indigenous practitioners to have more of a voice and a structure and a say in Hawaiian health society. And some potential solutions include cultural competency training for physicians, so giving physicians a baseline training before they go into the workforce. The establishment of Native Hawaiian health departments within the hospitals and clinics, so giving patients the option to visit this particular department, these people who are there for them and can help them connect to the healers and connect and refer to different practitioners that can help them specifically with what they want in a culturally sensitive way. And lastly, develop an organization of Hawaiian healers as an entity that the public health system can communicate with and that the patients can refer to as well. So some conclusions in terms of the needs and the next step to follow. We need to restructure our public health system to meet the needs of the Native Hawaiian community for both healers and patients. We need to emphasize cultural competency for Western practitioners working on Native Hawaiian land with Native Hawaiian people. And we need to focus on holistic health and medical integration when we're working with these communities. So, some acknowledgements. Peter Vitusik for guiding me on this journey in Hawaii academically and personally and launching me into this world. And Phil Durham for starting my whole interest in environmental health and human health connections 4 years ago and helping me make sense of the whole research right now. And Noah Lincoln for inspiring the whole project by talking about his childhood with Native Hawaiian healers and his experience in the Native Hawaiian community. And Woods Institute for funding, Stanford Storytelling Project, and School of Earth Sciences for their financial support as well. And yeah, and Hawaii, because Hawaii gave me that opportunity, the people and the land, and I'm really grateful. And I also have, in the theme of gratitude and protocol and tradition, I have another little Hawaiian chant that I wanted to share with you guys, if you'll let me. I'm not over time?
Please do.
Okay. Uholaia kamakaloala, puaike alohala, kukaiia kahaloala, pavehi mai na lehua, mai kaho'oku ia kahalavaila, mahalo ena akua, mahalo ena kupuna la'ea, mahalo me ke alohala, mahalo ke aloha la. Mahalo.
Nicole, thank you for such a provocative and interesting presentation. I am going to open it for a couple of questions before we toast all of the speakers today. Norm, you had a question?
Just kind of a story from my parents' time, and I'm not sure whether this came into Western medicine from traditional Hawaiian medicine or not, but if you get jungle rot, which was very common in World War II, it was known that you got up above the trade wind inversion, and then the dry air would go away very quickly, and this was used in World War II, and I presume the Native Hawaiians knew that, but I don't know for sure. I just kind of a
Jungle rot?
jungle rot. You get
Is that?
just in the heavy humidity of the rainforest. If you get cuts or scratches, they get infected and they get fungus in them and stuff, and it's very unpleasant and eventually becomes debilitating. But if you just simply go up to high elevation above the trade wind inversion, like on Mauna Kea, where all the cabins are, which is why the cabins are there. The military built them originally for this, and it just goes away because the fungus can't live in the dry air. And I don't know if they used that or not, but it seems something that the people would have known.
Yeah, I haven't heard about that specifically, but I have heard about differential rates of fungus growth along the elevation of Mauna Kea through Peter's biology classes. But I also do know that Native Hawaiians have a lot of remedies that involve, like, I don't know exactly elevation, but a lot of different types of plants they'll be able to use. And they're actually being really innovative in the way that they use it right now. Like, one of the most interesting ones I learned about is traditionally you use noni juice, maybe you use the poultice, like the pulp of it, and rub it against something or use the leaves. But right now they're even using like a nebulizer to inhale it, and they're trying to like combine Western technology with traditional methods and really trying to reach out to the people. I know this isn't supporting exactly what you said, but yeah, they have a lot of really interesting remedies.
Real active ingredients.
Let me open it to another question. Caleb,
Go ahead,
question? Yeah, I had a quick question on how, with this like focus on like, you know, your spiritual life is very crucial to your physical health. Do Native Hawaiian communities deal with like birth defects or certain diseases that like Western medicine would be like, that's just unfortunate? Are you kind of morally judged?
It's really interesting. That conversation actually has come up, and for them, it's about embracing everything and accepting and just constantly being happy with what the world gives you, which I personally thought was a great outlook on life. And the way that they see problems coming, you know, just coming in, they decide that they are going to accept it and like learn from them, and that your mistakes are, you know, your biggest problems, and that like people, the way that they're born, are natural and beautiful. And a lot of them actually don't even want to go to the Western healthcare system for births. Like, they don't want to take any sort of medication to speed up the birth or not feel the pain, and they choose to have natural birth. And they know that there will be consequences, but they prefer to be as natural as possible, and whatever is born in all the issues that we do have or may have, like, that's how the spirit meant it to be, and they accept it and it's natural.
Let's have one more question, and then I'm going to turn it over to our Dean, Pam Matson, to toast the students. Yes.
I want to ask a deliberately naive question. You make it sound so logical, so convincing. The data make it clear. Even the Western doctors agree that they should have more cultural competency. Why are we stuck? Why has it not happened what you're interested in?
Yeah, I think that goes back to the second set of hypotheses that I really have, that it's ingrained in like the fundamental structure that we have right now, and that's something that needs to be transformed in order to give the doctors the capacity to learn what they want to learn and be able to help the patients. So,
are doctors so many sheep? If they want to change this, why can't they change it?
Actually, I have talked to a lot of frustrated doctors about that specifically, and they said, you know, they don't have time to learn about all these things or like go beyond and get involved in the health policy because they're really busy in their day-to-day work schedule, and it's like loaded as it is. She's like, I don't even have time to exercise and carry out my life. Like somebody else needs to come in and do something. So I think that responsibility lies heavily on the Hawaiian health government, like public health arena and policy initiatives. And I do know that there's a lot of people working towards that. In the School of Public Health, for instance, like there's a whole new program focusing on Native Hawaiian health. And there's a lot of seeds right now of initiatives, but I think the importance of all of these initiatives hasn't been as widely recognized as it should be. And that's kind of what I'm trying to do.
I know there's a lot more questions for Nicole, so I encourage you to continue the conversation.
Well, let me grab your attention for just a moment, just to draw a conclusion to what I think has been an absolutely wonderful day. I wasn't here for all the talks, but the ones I heard and listened to outside and read about in here are amazing. But I know for you guys, this is not just a day, this is not just a presentation, but this is a whole heck of a lot of work that went on for a long time. So how does it feel to have done it? I mean, quite an experience, isn't it? Isn't it just an amazing experience to put that much work into something from the very beginning to the very end, and to actually be the person in the room who knows more about it than anybody else does. And that's what you guys have done. You've basically been transformed from students who are receivers of knowledge to students who are producers of knowledge. You've been transformed from students who are learning from other people to teachers who are teaching the rest of us what you've learned and what you now know about very deeply. So that's a pretty exciting transition, I think, in one's life. So congratulations for that. So we want to toast the students who've taken on this really incredible challenge. It's a huge intellectual challenge, and I'm sure at times was stressful and hopefully at times a lot of fun. Here's to you for teaching us so much about a diversity of topics, a whole different range of things. It would have been fun to sit in the room and learn from all of you today, as I know some of the folks here did. And welcome. You're on the next generation, a new generation of scholars, of people, researchers who study, learn something about how the planet works, how our systems, how people and their environment work together. But you're also, I think, a new generation of leaders who will use at least some of that information to make a difference in people's lives, to have an impact on the future. So congratulations for that too. And then finally, I think we do also need to say thank you to the mentors, your families, your friends, Richard, Saracina, Dina, I mean, Julia, all kinds of people have made a big difference in your lives in this process, many more than I know. And I think without them, of course, it would have been very hard to accomplish what you've done. So to all of you, here's to a wonderful conclusion to a very challenging set of studies and a very challenging time, and on to the next things. Congratulations.